Hello students, are we live? Come on, yes, we are live. Are we live? Quickly let me know in the chat box. Yes, we are live. Yes, hello, am I audible? Good evening, good evening. So we are live, the wait is over, my wait is over. Hello, hello, good evening, good evening. Yes, quickly like the session, and let's mark your attendance. Yes, yes, hello, hello. Good evening, good evening. Did you all mark your attendance? Did you all mark your attendance? I cannot see the number. Yes, Avengers assemble quickly. Share the link with all your friends. Ask them to join quickly and let the chats be flooded with fire emoji. I am looking at your chats. I'm going to answer all your doubts. Okay. And we have some disclaimers also. Like how movies have disclaimers. No, I have a disclaimer for this movie. This is longer than a movie, right? Yes, okay. So the energy is amazing. I want all of you to share the link with all your friends because many are still lazy. They have not joined, right? This is Game of NEET. How can you not be a part of it, right? Are we all ready? Yes, okay, great, 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 great. When I am checking your chats. Okay, so students, we have reached day 5 of Game of NEET, okay, and if you have completed all the 5 days, that means all the 4 days, right? Today HSP sir's class was their special class, so if you have attempted all of that, that means you have completed 4 chapters thoroughly with the practice, and now we are going to enter our 5th day and our 5th chapter, which is an amazing success for all the students because I am sure 90% of them wouldn't have taken part for it because thinking that why will I watch such a long lecture, right? But if you have done it, I hats off to you all, okay? Hats off, okay? From my side to all my future doctors, okay? So students, disclaimer for the movie is this much. There will be spammers, okay? There are going to be people who are going to talk to each other as if WhatsApp does not exist, Telegram does not exist, Instagram does not exist. So I want all of you to block them and be a part of the class. Don't go on replying to them, did you eat food, did you drink water, all that not required, okay? And I will be taking care of all your doubts because your chats are going continuously. I am not, I cannot focus suddenly, okay? But I will definitely give you a point where I will tell you students, if you have any doubt, please put in the chat box. I will read all your doubts. Clear to all of you? And all my students have the biggest confusion, ma'am, will you give break? Yes, I will be giving you break, mostly 1 hour, okay, 1.5 hours. After that, we'll have a 5 to 10 minutes break, okay? Yes? Okay, did you all share the link? Did you all mark the attendance? I cannot see the attendance. Come on, don't be conjuice. Just put a like. Just put a like. Yes, you can take running notes because I will be writing running notes with you. So, my speed is slow because I have to do decoration, I have to use colors. So, by then you can finish writing, okay? The slides are not ready made. Everything is empty. I have to start writing. Okay, shall we start? Shall we start? Yaar Vijay, I am standing here, you are asking about another teacher. I am standing here, be happy, no? Okay, so students, can we start? Ready for day 5? Yes, I will be posting the notes. I will be posting the notes. Yes, amazing, amazing. Okay, so let's start. So our topic is excretory products and elimination. Students, since you know that the syllabus is reduced, we are expecting up to 16 marks from this chapter. So at the end of this life, many will ask, ma'am, is this enough? Yes, this is enough. I have included every single word of your NCERT. So you do not have to worry. Just watch the complete lecture. Okay, I will also clarify your doubts. Everything in NCERT is on this PPT. Okay, that means whatever I am going to write. Okay, so I can guarantee you 16 marks at the end of this. I don't know how many hours this will go. Duration, I am not sure. Okay, so at the end of this session, you will be for sure getting your 16 marks. Clear? Okay, clear? Are we ready? Yes, 16 marks will be in your pocket. Exactly, 16 marks in your pocket because they have reduced the syllabus. Chances are high that these topics have come. And students, we all know our favorite, this channel has got the maximum love from our Tamil Nadu, and we know Tamil Nadu is going through a very bad phase. We have lot of students from there, and we are wishing all of them to be safe. I am wishing all of you to be safe. Okay, preparation is the next thing, but first be safe. Okay, I hope your family, your relatives, everyone are safe. So we have some surprise. Your Avengers teachers are coming to Chennai. Okay, so this is what all you have to know. You have to fill a form that is already put on the Telegram channel, and these educators will be coming to Chennai to visit you all. Okay, so if you want to be part of that journey, quickly go fill the Google form which is on the Telegram channel. Clear? Okay, so all of you, yes, you are all visible to me. You are all visible to me. Okay. Yes, Ambika ma'am is here. Are you all happy? So students, Ambika ma'am, HSP sir, Wasim sir, Kailash sir and Pankudi ma'am will be visiting you people in Chennai. Okay, so I hope all of you will be happy to see your top educators with you nearby, right? Nearby you can visit. It's not anymore through the screen. So quickly go fill the Google form so you can be a part of this journey. Clear? Right? Yes, okay. When will I meet? I will be like the big boss wildcard entry. Okay, I will come wildcard entry. If you people tell Gopika ma'am is needed, then I'll come as wildcard entry. But not as of now. Okay. Yes, so let's start the lecture. What are we waiting for, right? Students, your board shouldn't give heart. Okay, students, and we also know about the Great Learning Festival. First time ever, they have given a complete trial free to know how your plus and iconic batches work. So what are you waiting for? It is for free. Okay, just put your phone number, put OTP, put activation code Gopika and join and see how this plus classes work, how your plus educators teach, how are this doctors teaching, right? There are a lot of doctors in part of our journey who are teaching really well for students, who have given ranks in your all India ranks of 7, 9, 10, 11, 12. Okay, so you can also be a part of this. Just use the code Gopika. Okay. Yes, I will also join. Thank you so much. You people are asking for a wildcard entry. Yes, but this is not the Josh I expect. If I have to come, you have to show more Josh. Then we will do. Okay. Yes, let's start the class. So students, excretion. Okay. When we talk about excretion, I hope you all know that in digestion, we studied, digestion is completely removed from your chapter. In digestion, we studied that the unabsorbed, undigested food is getting out, right, in the form of feces. Right, that is removed out completely. Now, why are we studying excretion? Why? We study digestion, right? Why are we studying excretion? No. So, here in excretion, we are mainly focusing on the liquid waste. What is our liquid waste? Urine, right? We are mainly focusing on urine formation and our liquid waste. Okay, so this chapter is completely focused on what are the excretory products and how are they eliminated out of our body. Okay, so normally how does our excretory, the whole system looks like? It will have two kidneys, right? It will have two bean-shaped kidney. All of you know it will look like a rajma, right? And then it will have a ureter, right? And it will have a triangular urinary bladder and a urethra, right? This is a normal structure. Yes or no? I want all of you to draw and I will ask you to post your notes on the Telegram channel. You have my Telegram channel which is Biology by Gopika. Okay, join the Telegram channel. I will ask you people to post your notes there. Okay, so don't think and watch. Don't think this is a movie and sit and watch. I want all of you to write. Clear? Did you all mark your attendance? Yes, okay. So students, this is your kidney, ureter, urinary bladder. Okay, so this constitutes your general system, right? This is all about it, right? But this is all about it, but you need to know a lot about this. Am I right? Urinary bladder, and here we have urethra through which the urine will be expelled out, right? We have the urethra with which the urine will be expelled out. So, we are talking about excretion. When we talk about excretion, we are talking about the liquid waste. Now, all of you tell me in the chat box, what is excretion exactly? If I have to tell a person who does not know anything about biology, I have to give them a very small definition. What is that definition? Quickly put in the chat box. give me the definition of excretion. All of you, quickly. What is the definition of excretion? Very good, very good. Everyone type in the chat box. You don't have to be like, oh, I'm first time to this ma'am's class, I don't want to type. Nothing like that, no? Those who are first time to this class, you people should put the fire emoji first, okay? You people should put the fire emoji for not attending my this many classes, okay, as a punishment. Very good, very good. Removal of nitrogenous waste. Removal of nitrogenous waste. Now, how does this nitrogenous waste get formed? Right, it is formed because we are doing other metabolic activities, correct? So, due to metabolic activities, this nitrogenous waste is expelled out. Exact. Very good, very good. All of you have put the definition. So, if I have to tell a layman what is excretion, I am going to tell removal of nitrogenous waste. Now, students, such a big organ. 2 kidneys there, 2 ureter is there, 1 urinary bladder. They will do only excretion. Will they do only excretion? No, they will also do something known as osmoregulation. Okay, they will also do something known as osmoregulation. Okay, so what are they going to do? Osmoregulation. Now, all my students have studied osmoregulation. Yes or no? Such a big organ cannot do just excretion. And we have studied in animal kingdom, whoever is doing excretion is also going to do osmoregulation. So, osmoregulation means salt and water balance, right? Yes, very good. Salt and water balance. Salt and water balance. How many of you, as soon as you see question paper, your BP will go low, right? As soon as you see question paper, BP will go low. You'll be like, I'm getting headache, I'm going to fall now. Standing in assembly, everyone will have BP low. One will fall, next person will fall, next person will fall, next person will fall. Yes? Yes? Biology by Gopika. Jason, biology by Gopika. So, in assemblies, one time at least you would have fallen in your life. Why? Because your osmoregulation wasn't proper. That means your salt and water balance wasn't proper, right? You will be in competition. Oh, that fellow fell today. Tomorrow I will fall. Right? Chumma, you're standing also, you will be like, ma'am, I want to fall. Right? So that you can escape from the first hour. And especially first hour is mathematics. Then definitely on that assembly day, you have to fall. Yes or no? Yes or no? You have to fall. Right? Maths hour I cannot attend, I have to fall today. So, you will force yourself to fall. That is not because of osmoregulation, that is because you did your osmoregulation, right? You started adjusting with the outside temperature, right? They are called as what? Osmoconformers, who can change their, osmolarity with the outside temperature, right? With the outside osmolarity, outside salt and water content. Got it, all of you? So, such a big organ is going to do osmoregulation as well as it's going to do excretion, okay? Removal of your nitrogenous waste due to metabolic activity, and it is also going to do osmoregulation, that is salt and water balance. Okay. Now, the question arises, what are these excretory, what are these nitrogenous wastes? Who is this people? Who are they? We need, right? India wants to know, who are they? Tell me, students, who are our nitrogenous waste? I am writing NW because my kidney is very big, I do not want to reduce the size of this. Okay. Tell me, students. Yes, very good, very good. Thank you so much. Lot of compliments also I am getting. Students, I will reply to everything, okay. You can put it in the comments also, I will reply, okay. Students, what are they? Ammonia. Okay, I think I have to reduce my this. Okay, let's remove this. Yes. Urinary bladder. What are these nitrogenous waste? Okay, these nitrogenous waste are nothing but, okay, we have ammonia, right? We have ammonia, we have urea, we have uric acid. these people are expelled out as nitrogenous waste. Are they the only nitrogenous waste? No, there are many other nitrogenous waste which we are not bothered, because we only stick on to our NCERT, right? There is lactic acid, creatinine, so many things are there. We are bothered about who? We are bothered about ammonia, urea and uric acid. Ready? Ready? So students, now that you have got to know what's the flow of my class, I want you all to share the link with your friends. Ask them to join. Tell them we have to crack NEET this year. Next year we don't want to see, we don't want to do this again. Okay. So quickly share the link. Okay. I hope all of you got enough time to write your notes also. Okay. So let's go to the next topic. I mean, not the next topic, but I'll teach you osmoregulation. Okay. Now I told you what is osmoregulation. Is basically salt water balance, right? Salt and water balance. Now we have to know a graph. Okay, we have to know a graph. Okay, so that graph is nothing but 1 second, students. I'll be artistic. I'll use tools. Okay, the line is also coming with me. 1 second, students. Yes, we have succeeded in drawing it. Okay, so students, this is a graph. Okay, this is a graph. This is 2 lines. Okay. So what are we going to do? So I'm going to write here osmolarity. Osmolarity means same here, osmoregulation itself. The mole, that means the amount of salt content in it. Okay. So osmolarity of the external medium. Okay, external medium, and here osmolarity of the internal medium. Okay, this is the graph all about. So we have x-axis, y-axis, osmolarity of the internal medium, osmolarity of the external medium. Now depending on who can regulate, right? I told you assembly example. You became osmoconformers. That means what? You regulated your osmolarity with the mathematics class, with the assembly, with your teacher giving you marks card, with your teacher giving you your answer sheet, right? That means what? You are able to change your internal factors depending on the external factors. Okay, those kind of organisms are called as osmoconformers. That means they change their osmolarity depending on the external medium. Is this clear? Is this clear? Right? Okay. So we draw them. Okay. They show a straight line. Why? Because external is changing, so internal also is changing. No tension, right? Outside is there. Outside students, okay, they're not studying. Even I will not study. No tension, right? So that is osmoconformers. Students, orange is visible, right? Okay, osmoconformers. Okay, now what do? If you want to write it, you can write change the osmolarity. Change the osmolarity depending on the external medium. Okay, on the external medium. Okay, then this example is very, very important. Okay, the example of osmoconformers will be smaller organisms, right? What are smaller organisms? Are invertebrates, right? Invertebrates. So invertebrates are the example. Some places you will also see invertebrates with mycene. Okay, mycene also shows osmoconformer behavior, even though it is a vertebrate, because it is a very primitive vertebrate. It will do osmoconformers. So if mycene is asked in your exam, do not hesitate to write it is an osmoconformer. Clear to all of you? Okay, clear to all of you? Yes. Boys, is clear? What happened? Some ambulance symbol and all you people are putting. Thank you, thank you. Students, voice is clear? All clear? Yeah, don't waste time and tell ma'am lag is there, voice is not clear, ma'am you're seeing is 4D, 5D and all. Don't tell to waste time. Okay. Yes, okay, clear. Fine, fine, fine. Thank you, thank you. Okay, fine. Okay, students, the number is not increasing. Okay, number is not increasing. What is this? You people don't have fans. You people don't have friends. Call everyone. Tell them I will buy you chocolate and give. Come and join. I don't know what they will ask for, okay? Yes, I am in Unacademy. I'm in Unacademy, okay? Yes. So, students, osmoconformers means change the osmolarity depending on external environment, and the example is invertebrates, okay? Invertebrates and mycene, okay? Now, we have us, okay? We have us. Can we do that? We cannot do that, right? We cannot do that. One second, I will just, yeah. So, we are, I'll draw it here, students, just to label, okay? We are osmoregulators. That means we cannot change our temperature, our osmolarity depending on the external. So, we are what? Osmoregulator. We are osmoregulator. Clear to all of you? Yes. Okay, osmoregulator. So, that means what? They do not change depending on the outside. Okay, I am not writing it here because it will become very clumsy. Please, all of you make a note. Okay, right? They do not change depending on the external environment. Okay, then we have some students who started game of NEET in day 1 and then lost all the energy. Same set of people we have here. Partial osmoconformers. I'll tell you why they are our game of NEET first set of students. I'll tell you why. Look here. What they will do, okay? They will come with lot of energy. Ma'am, I will complete game of NEET. And what will happen? At one point, gone. Have you seen those crackers? We will think that big, huge sound will come. But as soon as you light it starting, they will show that, oh yes, I have all the energy. Suddenly, it will be like, gone. Right? These are this, okay? So, here, these are partial osmoconformers, okay? That means they will have some time energy. Okay, I will do osmoregulation for some time. That is the straight line. After some time, they're like, nahi hoga, I can't. I will just do what the other people are doing, right? So, they are partial osmoconformers. So, students, I want you people to be osmoconformers. No, I want you people to be osmoregulators. That means whatever happens outside, okay? If I'm not telling if a bomb explosion is happening, don't go and bother. But if there are small things happening, do not bother. Okay, this time we will be in AIMS, right? We will be in AIMS, okay? How long this live will be, even I don't know. We both will find it out together. Okay, until I finish the chapter, the live will go. Clear? Clear? Yes. Okay. So, partial osmoconformers. That means they will do osmoregulation for some time, then they will lose their energy and they will do they become osmoconformers. Clear to all of you? Now, what are the examples? Osmoregulators, vertebrae is the example, right? Vertebrae is the example. Okay. Vertebrates. Partial osmoconformers examples are different. Okay. Elasmobranch. Right? All of you know your sharks and rays, right? Example, shark and rays. Okay, clear to all of you? Elasmobranchs. Okay, elasmobranchs are partial osmoconformers. Example is shark and ray. Is this clear? We studied the entire thing with just one graph. Okay, entire thing with just one graph. Yes, whole chapter I'll teach. Then what you expect? Half chapter me to teach, half chapter another teacher to teach. I have to only teach, no? Complete chapter. Yes. Okay, done? Can I? Okay. Mycene, have you seen your hagfish? Your, you have studied this in animal kingdom. Remember, all of you? Yes or no? Yes or no? Yes. Okay. So, this is all about it. Shall we go to the next one? Okay. Yes, students, add 1 second. Yeah. Okay. So, mycene, if you know animal kingdom, you will know it, right? Students, what is the other name for mycene? All of you tell me, what is the other name for mycene? Remember, we studied petromycin, mycene, right? We studied PM as the way to remember, right? That is, if you study animal kingdom, you will know that, okay? So, students, yes, Excretory products. I told you there are 3 types of excretory product: ammonia, urea and uric acid. So, we are going to study about ammonia. Now, students understand, ammonia, urea, uric acid are very, we cannot keep them in our body, telling that, oh, let it be in my body, nothing will happen. You will vanish if it is kept in the body for such a longer time. You will vanish if ammonia is kept in your body for a very longer time. So, what is the first sentence I can write? It is highly toxic. It is highly toxic because of which it has to be readily removed from my body. I cannot wait. Oh, a kidney will come, do the process, slowly excretion will happen. By then I will not be there. Okay. So, ammonia has to be readily removed and we will not take the help of kidney. Okay. Very important. Readily removed, okay, readily removed from the body. Okay. Do we take the help of kidney? No, kidney does not take part in this process. Okay. Kidney does not take part. Take part in what? Kidney does not take part in removal of ammonia. Okay. Yes, more water. Very good. Okay. It requires a lot of water. That is the reason we are not excreting out ammonia. Students, what are we excreting out? What are we excreting out? What are we excreting out? Tell me. Quick. Okay. So, kidney does not take part in it because, very good. We are excreting out urea. Does that mean ammonia is not formed in our body? Students, by the way, I have something to tell you people. In between the slides, okay, in between my whole slides, you will have quiz. Now, what is the quiz all about? You will have quiz with some diagrams given. You will have to label those parts. So, the quiz will go in such a order. Once I finish teaching that topic, then anywhere in the slide quiz will come. I don't know where the slides are. So, quiz will come and you people have to answer. Clear to all of you? Are you all okay with the plan? Yes. Quiz will come and you people have to answer. Okay, I don't know where it will come, but definitely after I teach that topic only it will come. Yes. Okay, great. And you have to keep your score of you have to know your marks, how much you scored. If 3 questions are there, all 3 correct, give yourself 3 marks. Clear? Yes. Okay, fine. So, kidney does not take part, we excrete urea. Now, does that mean we do not make ammonia? No, we do make ammonia. What is going to happen? The ammonia that is formed in our body, okay, will enter our liver. It will enter our liver and with the help of urea cycle, right? With the help of urea cycle, it will get converted to urea. This is in humans. Okay. So, got it? The ammonia that will be formed in our body will be entering the liver and in the urea cycle or your ornithine cycle, it will undergo, the ammonia will undergo or get into the urea cycle or ornithine cycle and get converted into urea, because our body cannot keep ammonia even for some time. Okay, clear to all of you? Okay, I am erasing this. This is just for your information. I will be telling this in your urea when I teach you urea. Okay. Yes, let us continue the points. It requires a lot of water, right? It requires a lot of water to be removed out, because of which why we cannot do it. Okay. And it is released out in the form of NH4 ions. Okay. It will be released out in the form of NH4 ions. Clear to all of you? Okay. So, and it is highly soluble in water. It is highly soluble in water. Clear? Okay. most toxic substance, right? Most toxic substance. It is readily removed from the body. We cannot wait for the kidney to come and do aram se excretion. By then person only will get vanished. So, we will tell kidney, you relax, you do what you can, we will remove it from the other process, right. So, kidney does not take part. So, it will be removed from how? It will be removed from body surface, okay. It will be removed, from body surface by diffusion, okay. It will be removed by body surface, okay. And no role of the kidney, I told you, requires a lot of H2O. We cannot give that much H2O, okay. We hardly drink water. In that also, if we have to emit out ammonia, not possible. So, we didn't take this risk. And if ammonia is there in our body, it will immediately go to the liver. It will do urea cycle, get converted to urea. We are safe, okay. It will be going out as NH4 ions and highly soluble in water. Clear to all of you? Yes, okay. Now, students, if an organism is giving out ammonia, what is that organism called as? If an organism is giving out ammonia, what is that organism called as? We are conducive to, you people are not only conducive to give water, you people are conducive to give likes also, comments also, share also. As if I am asking pocket from your money, you people are so conducive in doing everything. Everything you people are conducive, okay. Yes. So, the organism that is doing ammonia is called as ammonotelic, right. Ammonotelic organisms, and the process is called ammonotelism. Clear to all of you? So, the organism that will be doing, it will be called as ammonotelic, and the process is called as ammonotelism. Clear to all of you? Yes, good job, good job. Okay. Now, who are these organisms that are going to give out ammonia? It should be organisms who have ample amount of water to give it out, right. So, those organisms are our marine organisms, right. Marine, who lives in water, they are like, okay, I have enough water, I don't have to worry, right. So, let us write some examples. Sponges, they are always in water, right. Your marine or aquatic amphibians, aquatic amphibians, right. Your flatworms, okay. Okay, these are all the examples, okay. Even your bony fishes, let us add that also. Okay, we have bony fishes, aquatic amphibians, flatworms and yes, okay. Okay, good job, good job. Okay, so sponges, aquatic amphibians, flatworms and bony fishes. These are the ones who are capable of giving out ammonia because they are always in the aquatic mode. Okay, let us go to the next one, that is we are excreting out urea. Right. Yes, your flatworms also, you can write as an example, okay. Now, urea, when I talk about urea, what do I talk about urea? They are less toxic, right. I am comparing it with ammonia, okay. They are less toxic. Do they require a lot of water? They do not require they do not require lot of water, and so I will write they are less soluble in water. They are less soluble in water, okay. Clear? And in humans, what did I say in humans? In humans, if ammonia enters, okay, I will use a different color to write that, so you will know it is important. Ammonia is there in the body, it will enter, okay, your urea cycle, okay, in the liver, and get converted to urea. Okay, clear to all of you? Urea cycle, ornithine cycle, all these names are there. You can use any of these names. Okay, so ammonia will enter the urea cycle in the liver and get converted to urea. Clear? So, we are basically protecting ourselves, so we do not get any harm, right, because ammonia if in body, it is too harmful, okay, and it requires less water for removal. requires less water for removal. Okay. So far any doubts, students? So far any doubts? Yes, it is in decreasing order, correct. Terminology, psi, correct psi. Yes. No doubts? All clear? Can I get some fire emoji? Chat should be moving, okay? So I will not be able to see. Chat should be moving. Show the Josh, because your energy is directly proportional to my energy. If your energy goes down, my energy also will go down. Okay? Yes, it should keep moving. Yes, amazing, amazing. Wow, very good, very good. That's crazy. Nice, nice. Okay, so it requires less water for removal. Okay, wow, students, so happy that we have such an amazing set of students. Okay, such an amazing set of students. Okay, you people will crack AIMS, students. You will be all a part of AIMS. Trust me. Teachers' blessings are, teacher just by sometimes see, now I feel very emotional and happy seeing all your chats. Even by that emotion, if I tell you people will crack AIMS, no, that blessing will come. So don't worry, all of you will be in part of AIMS. All of you, I pray that all of you are not, if you don't want to be in AIMS also, if you are deciding of some college, I hope all of you are there. Okay. Yes, you will all be in AIMS Delhi, do not worry. If not in AIMS Delhi, other places, whichever you want. Okay, let's start. So requires less water for removal. Okay, now we have to write example. Before example, what we have to write? We have to write, what are they called? What are the organisms called? What are the organisms called if they are releasing out urea? Yes, yes. Okay. So they are called as uricotelic organisms, and if they are, that process is called uricotelism. Uricotelism. No, one second. Ureotelism. Ureotelism. Right? Ureotelism. Students, uricotelism is that uric acid. Okay, don't get it confused. Uricotelism is your uric acid, right? So ureotelism. Ureotelism. Correct, correct. Most of you have got it right. Here and there, key sounds problem will come, but urea and telism. Okay, right. Example. Let's write example. What are the examples? We are the examples. We are the examples. Correct. We do not give that much of water. So first we will write adult amphibians. That is your frog. I am writing this specially because you remember when we studied frog, we had specially mentioned they are uricotelic organisms, right? Uricotelic organisms. Very good. Mammals. Okay. And cartilaginous fishes. Yes, cartilaginous fishes. Okay. Yes, great. Okay. So adult amphibians, mammals and cartilaginous fishes. These are the organisms that give out urea as the waste material. Okay, waste material. Now the last one, the least toxic. The least toxic person is our uric acid. Okay, who is our least toxic person? Okay, tadpoles will come where? Tadpoles will come in your ammonia, right? Tadpoles will release ammonia because they are in water. Okay, Shyam, if I am reading your name right. Okay. Okay, next one is uric acid. Uric acid is least toxic, right? Least toxic. Least toxic, and it hardly requires water. That means it requires very less water or no water at all. Okay. Requires very less water. Are you all writing notes? No water at all. Okay. All of you are writing notes? Yes. Okay. And least soluble in H2O. So if they do not require that much water, it is understood that they are also least soluble in H2O. Yes. Now, students, all of us, once in our lifetime, it is like getting polio. Once in our lifetime, the bird would have thought, let me release some pellets on this person who is walking so stylishly on the road. Correct? All of us at least once in our life, like how we get injections for how we get polio, no? Once at least the bird should have done some kind of kripa karune on us by releasing some uric acid. Yes or no? Yes or no? You should be honest, okay? You shouldn't be like, no ma'am, I've never seen a bird. Bird has never come near me. What are you talking? Yes, no, correct. Because birds will be like, oh, this person is walking with so much style. Now I have to do something, right? Okay. If it hasn't happened to you, I am so happy for you. You people are one among, what is that, one museum species. We can keep you in museum. Okay. No ma'am, promise and all they're telling. Okay. Exactly. So that semi-solid is this uric acid. Okay. So whenever I go out, it happens that the bird will think, yes, this is my target for today. Okay. And I'll be walking. So when I'm walking, suddenly I'll decide to sit or to move or, to fix my sandal, something I will do. That the friend behind me will be the target. The bird's target is me definitely, but I will do some kind of circus that the target changes, right? Correct? So, my friends tell we should not walk with you. We should not walk with you. Yeah, people tell it is a blessing, but what blessing could it be that, you don't even realize, and then when someone tells you, you touch like this and you will be like, oh my god, who knew that it was uric acid? We all used to curse birds, right? Okay. Yes, so this is an easy way to remember it, that it is your pellet form, semi-solid form, okay, semi-solid form, okay. So, released as a semi-solid. This is the only way I can make you people remember. Semi-solid waste, okay. Right, that pellet, yes, very good, okay. If you people have escaped, very, very smart of you, you should escape like me. I don't know, somehow I suddenly decide to do something crazy there, so that the target moves and the bird is also disappointed. So, bird has not tried once, bird has been, I have been a target for a very longer time, but somehow in Bangalore, birds numbers have decreased because of the pollution and everything. So, I am, saving myself and going around in life. So, I thought after teaching, after spreading knowledge, maybe the bird will be like, oh, thank you, you made me so famous. So, I am not the target anymore. This is all a joke for you to remember, okay. So, because in exam there will be 100 things to remember, suddenly if you forget what is that uric acid form, you will remember this joke. That is why I am telling those students who are very serious in the class, why is ma'am cracking jokes and all. If you are telling students, everything that a teacher tells in class somehow will come to your exam. I am telling you, somehow you will remember, and I am telling you, our brain works in such a way that jokes will be remembered for a long time than all these notes, okay. Okay, so released as semi-solid, it will be in a pellet form, right? It will be in a pellet form, right? And who secretes it? Do we even have to ask? But the most important thing is why we do not form uric acid when it is the least toxic, when it does not require any water. We are not forming it because it is the most expensive product. Now, what do I mean by most expensive? Am I talking about buying it from Amazon? No, I am not talking about that. I am telling most expensive in these kind of things is what they use a lot of ATP, right? They use a lot of ATP. Okay, so while giving it out, the energy utilization is too high. That's why we are not emitting out any uric acid. Clear? Clear? Okay. Yeah. After this, I will try astrology. What and all you people want me to try, I'll try, students. No problem. You people study more than enough. If you people study, then I will try all the other kind of circus you want. Okay, first you people study, make this video viral, tell that this ma'am is teaching amazingly she's doing a good job. Make this video viral, then I'll do astrology, whatever you people want me to do. Okay, so it is the most expensive product because ATP is utilized highly and we cannot afford to utilize our energy for all these things. Okay, okay, all these things. Now give me the example. Give me all the birds that give you blessings. So shall I write the whole family? No, wait, we will go in order. We will first write the process, then we will go to their family, right? So what is it called? What is the process called? Yes, yes, correct. What is the process called? Urecotelism. Urecotelism, and the organism is called uricotelic and process is called urecotelism. Okay. Now we will go to our interesting part. Examples. Examples, aves. I'll write aves as a whole. Okay, the whole bird category we are going to write. Okay, but we will remember crow specially, right? Always in our hearts, crow will be there. Yes, aves. Not only aves, students, some insects, even some reptiles. Okay, or most of the reptiles. Okay, clear to all of you? Yes. Now the question comes. Okay, now there is a question. Do we humans make uric acid? Do we humans make uric acid? Yes or no? Yes? I am reading your chat and smiling. Don't think I am looking somewhere and smiling. I am looking at your chat only. Oh, that's nice. No, no, okay. All of your answers is no. But let me tell you, all of you are wrong. I'll tell you why. Humans make some amount of uric acid, but that uric acid is not coming from as nitrogenous waste, but it is coming from metabolism of purines. What did I say? Small amount of uric acid is formed in our body, but that is coming because of metabolism of purines. Okay. Shall I write it here? Okay. Some amount, okay, humans form some amount of uric acid but that is coming from metabolism. It is not because of our regular thing, it is because of metabolism of purines. Okay, clear? Yes? Yes, metabolism of purines. Oh, 2 people told the answer. Good job, good job. Many people told yes actually. Okay, those who told no, this is a new knowledge for you. Please understand. Okay, humans form some amount of uric acid because the metabolism of purines. Clear? Aquatic animals, Sanvi, most of the aquatic animals will release ammonia because they have a lot of water in them, right? They have a lot of water in them, so they can eliminate. Now, specific organisms, if you go check, some might have exceptions, some might not have exceptions. Clear, Sanvi? If you want to know specific organisms, you have to search for it. Otherwise, generally, what do we say? Aquatic organisms will give out ammonia because it requires a lot of water and they also have a lot of water. Clear? Yes. 1 second, students. Students, purines, pyrimidines, remember our DNA nitrogenous bases? Purine is a type of a nitrogenous base. Purine means your adenine and guanine, okay, at Gopika ma'am's class, right? So, metabolism of purines means is a type of a nitrogenous base, okay? Yes. Got it all of you? Purines, yes, pure as gold, very good. Pure as gold, that is your P stands for purine, A stands for adenine, G stands for guanine. That means it is a part of our DNA. DNA has 3 components. In that, one component is your nitrogenous base, that is divided into 2, purines and pyrimidines. Okay, if you haven't watched my molecular basis of inheritance, go quickly, don't watch it now. After this class, go and watch. Okay? Yes. Yes, you will learn it in biomolecules, you will learn it in molecular basis of inheritance. Okay? Now, students, one more question. Is all the urea formed in our body given out? Is all the urea formed in our body given out? Yes or no? Is all the body urea formed is given out? No, very good, very good. It is not given out. Why? Because we need to do something called as osmoregulation. So, this is a very important sentence. So, I will put it in a box for you people, so people don't forget. Students, okay, I am writing it here only. Urea is not completely given out. No, if I write like this, space should be in. Urea is maintained in our body. I'll write it that way. Urea is maintained. Okay, 1 second, students. I'll write it this way. Yeah. Some urea is maintained in our body for osmoregulation. Clear? Okay, please make a note, put a star and write it, okay? Yes, yes. Thank you, Jasmine. Okay, so students, did you all like the session? Let me see the attendance, okay? I'm a very strict teacher. I'm going to check your attendance. You cannot bunk my class. I'm going to check the attendance. If few people haven't liked, quickly like. By the time I open, you have time. You have time till that. Good job, good job. Good job. You people are saved because you people have liked. Yes, more likes should come quickly. All of you like? Yes, good job. Okay, so students, this much is clear, no? Some urea is maintained in our body for osmoregulation. Very, very important. It was asked as an MCQ. I don't know which year exactly, but it was asked. Okay. So, when we were doing this MCQs, I saw this question and I was like, oh no, I have to inform this to students. So, please make a star and write it big capital letters in your notebook and keep, because before your exam, you will not know what to revise, what to do, and you will miss this one line. Okay. Remember this and keep. Now, students, we have to do a quick revision of our animal kingdom. What did I say? We have to do a quick revision of our animal kingdom. Okay. I will give you ample amount of breaks. Okay. So, do not cry. Ma'am, break. Do not cry. I will give you after 1.5 hour. 1.5 hour, I will give you 5-10 minutes break. Okay. Thank you, Kaustuba. Yes, we need more students. Lazy, NEET aspirant. Bring in more people. All the lazy people, you make them awake and bring them. Students, why urea? Why urea? You will get to know when I am teaching you about your RAS mechanism, when I am teaching you about your urine formation. I will tell you why urea is maintained for osmoregulation. Okay, clear? Okay, clear? Yes, fine. So, students, let's do a quick revision of your animal kingdom. You have to tell me. So, this is your first quiz, okay? Not your quiz, I will be helping you for this, but then later quizzes I will not be helping you. Now, you have to tell me platyhelminthes, annelids, cephalochordates and all. What are the excretory organs? Come on, quickly. I will be seeing your chats and I will give you your scores. Okay, you have to tell me what are their excretory organs. Start. Yes, Shailu, very good. Very good, Chanel, Ruth, Kanishka, Mishra, Subhashni, Kaustuba, Nikila, Lijo, Praveen, Pavadharni. Very good. Yes. Oh my God, so fast. Now I cannot see students. Susan, Vinny, Lazy Aspirant, Karuna, Laharika, Sai Naidu, Jason. Very good. Protonephridia and your flame cells, right? Flame cells, right? Now, students, when we studied, right, when we studied about these organisms, we also studied that these people help for excretion as well as osmoregulation, right? Remember when we studied flame cells, there was a line in your NCERT that they help for excretion as well as osmoregulation, correct? So, here that is the same thing, we are connecting our humans also. Our whole excretory organ we are connecting with osmoregulation. Next one, earthworm. Earthworm, did you already people put the answer? Yes, very good, very good. Nephridia. Earthworm. All of you have revised animal kingdom well. Nephridia, right? Earthworm is Nephridia. Now insects. Yes, insect. I think someone already put the answer. Yes, very good. Nephridia, correct. I have written protonephridia, flame cells, to make you understand that in some place, see in annelids, if you see, you will see flame cells, right, in the NCRT. So I have put both of it. Okay, nephridia, Malpighian tubules. Very good. Malpighian tubules. Yes. Okay, last one, your prawns. Prawns, Malpighian tubules, correct for insect. Remember, we studied it in our arthropoda. When we studied, we understood that the excretory organ was your Malpighian tubules, right? Yes, very good. Green glands. Green glands. Okay, good job, good job. Green glands. Yes. Antennal cells, green glands, both you can write. Okay, both you can write. Both the answers are correct. Okay, antennal cells, green glands, both is right. If green glands are easier to remember, go for it. Antennal cells are easy to remember, go for that. Okay, so this is your one paragraph in your NCRT. Okay, so if you see your textbook, in this side of the top they have given, let's revise animal kingdom, and they have given all of this. Now how is the question expected? The question will be in the form of a match the following or incorrect. Okay, they will give you the incorrect pattern, you will have to select the correct pattern. Clear to all of you? Clear to all of you? 4-mark question in this one box. 4-mark question. Okay, so you are going to get your 4 marks if you know this box. They are going to incorrectly give the order, you have to correct it. Clear? Yes, good job. Fine. Now we are going to start. Our actual storyline is over, students. This was the teaser and trailer is over. Now we are going to get into the story. Yes. So our story starts here. Human excretory system. Right? Human excretory system. What and all is there? Movie starts now. What and all is there in our human excretory system? Kidney is there, ureter is there, urinary bladder is there, urethra is there, right? All these people are there. Okay. Now you have to give me time until I draw the structure so I can explain it to you. By then I want all of you also to draw it. Okay, I want all of you also to draw it. So we will finish it at the same time. Yes. So I have done the basic structure. Okay. Okay. Yes, the basic structure here it is little, so I'll just erase off this portion. I'll just erase this because it becomes too accumulated. I cannot explain all of you. Yes. Are you all done drawing? Okay, done? Yes. That's my kidney, ma'am. Okay, fine. You want to take my artistic, this one you can take, no problem. Okay, students, tell me, tell me, one kidney is lower than the other kidney. Who is that kidney? Don't send me your diagrams. I don't want to die in such a young age. If you're sure that I'm going to get heart attack seeing your diagrams, please don't send. Okay, so tell me students, which kidney is lower? Your right kidney is lower than your left kidney. Why? Okay, so left, so 1 second, yes. Your left kidney is a little above, okay, little above, and your right kidney is a little below. That is because the liver is pushing the right kidney a little down, okay. Liver is pushing the right kidney down, okay. So, my right hand, so this one, liver will be pushing this down, clear? Yes. So, the reason you have to know that the right kidney is pushed a little down because of the liver. Very good. Okay. Now, students, we know that artery and vein is entering the kidney. All this you people know. Now, you people have to know that the renal artery is going to carry oxygenated or deoxygenated blood. Renal artery is going to carry oxygenated blood. But the question is the oxygenated blood lacking any toxicity? I am talking in the terms of toxicity alone. I am not talking in the terms of oxygen or carbon dioxide. You people have to tell me, is the oxygenated blood that is coming in, is it completely pure, no toxicity at all? Yes or no question. If you are telling yes, that means you are telling ma'am toxicity is there. If you are telling no, that means no toxicity is there. Now yes or no, no confusion, no. Yes, Mishra, very good. Okay. So, that means the oxygenated blood that is being carried by your renal artery, right, the renal artery is carrying oxygenated blood. Renal artery is carrying oxygenated blood. But the oxygenated blood is not completely void of toxicity. It has some amount of toxicity because of the presence of urea. Okay, because of the presence of urea. Yes, very good. Most of you understood it. So, you should understand that oxygenation we are talking depending on your oxygen and carbon dioxide. Okay. Now, when renal vein is bringing its blood, it is going to be deoxygenated. But in that deoxygenated blood, we will make sure that urea is not there. That means that blood will not have toxic substance. Clear to all of you? Yes. Okay. Great. Okay. This is a very general information I wanted to tell you because again I saw an MCQ. Okay. Let's now label our this artistic drawing. Okay. Let's label. All of you also label your diagrams. Okay. I am erasing this since you understood. Oh no. Kidneys portion. Yes. Let's label, students. So, we already know kidney, okay, ureter, urinary bladder, urethra, very important, urethra. And students, at the tip, okay, at the tip, I am going to draw 2 circles. You people have to tell me what is that. I am going to draw 2 circles. I am going to extend and draw, okay, so I can label. Tell me, I am drawing 1 circle, 1 more circle. These circles which I have drawn, okay, they are called your sphincter muscles, okay. There are 2 types of sphincter muscles, so I am telling S1 and S2, okay. Those 2 are sphincter muscles. We have all learnt about sphincter muscles, right, but we have 2 sphincter muscles, sphincter muscle 1 and sphincter muscle 2, okay. That we will do in depth, okay, that you are going to study again in depth, okay. So, this is you people are sure about this part, kidney, ureter, urinary bladder, urethra, you have your renal artery, right, we have our renal artery and we have renal vein. This is your human excretory system. Now, what and all this excretory system is going to do? It is going to help in urine formation. It is going to help in urine formation and it is also going to help, okay, in osmoregulation. And is it going to only help during normal conditions? No, it is also going to help during stress conditions. It is going to help the body during stress condition. Now, what do I mean by stress condition? When some person is fasting, right? When some person is fasting. We have, I am not drawing adrenal gland because we are not giving importance now. Otherwise, it will be a cap like structure like this. Okay, it will be like this. Clear? Like a Christmas papa's cap. It will be a cap like structure like this. Okay, adrenal gland. Because we are not bothered about adrenal gland. Why will you talk about someone who is not in this class? Correct, right? Now your, for them that satisfaction ki ma'am missed something, I have to tell, right? I want to be the hero of the class, ma'am cannot be. Okay, so adrenal gland happy, complete. Kidney, you kept a cap for the kidney. Okay, now students, so sphincter muscles clear? Okay. Yes, very good. They come under endocrine gland. Now students, what am I talking about stress condition? When a person is undergoing fasting, when you are sitting in a 5 hours lecture continuously. Now I will give you breaks, you cannot give my lecture as an example. Okay, imagine you are sitting in a offline lecture where the teacher is very strict and you are very scared to even ask them for washroom. You are sitting in a very constrained situation. Your body is going to regulate in such a way. Okay, you haven't drank water from morning because you are very tired, you're crying for something, you haven't drank water but you're losing out continuously water, that situation, right? So at extreme situations, body, the kidney and everything will regulate itself. Clear? Okay. Okay, clear? Fine. Now let's understand kidney. Okay, again we are going to get into our main hero. Okay, main hero. Okay, got it? Wow, you people are saying random. Yes, I told if you are scared of a lecturer, I never told that because the lecturer is boring, you are sitting in that class. No, no, no. Okay, yes. So let's start about kidney, students. So kidney is a retro yeah, 1 second students, I'll write it down. Too up, I cannot even see what I'm writing. It is a retroperitoneal organ, or it is present in the retroperitoneal space. That means what? Now what does this retroperitoneal mean? So students, imagine, okay, this is a circle. Just imagine this is a cavity. Okay, this is a cavity, right? And now I am going to draw the kidney. Okay, this is your 2 kidneys. Okay, this is your 2 kidneys like this. Now what is happening? Peritoneum will cover all the organs. Okay, peritoneum will cover all the organs. You would have studied this more in depth in your animal tissues, or otherwise also you would have studied. Now what is this peritoneum going to do? It is going to get covered. It is going to cover like this. Okay. Oh, this has been a 1 second. Yeah. So peritoneum will come like this. It will cover the kidney in such a way. Okay, it will cover the kidney in such a way. Are you all seeing? Now the kidney is attached. Where is it attached? Now if I have to label this, this is the ventral side, this is the dorsal side. The kidney is attached to the dorsal side. So what has the peritoneum done? The peritoneum came, it covered the kidney on the ventral side. It did not cover the kidney on the dorsal side. Why? Because it is attached to the dorsal side. Okay, so this very important thing you have to understand, that the kidney is attached to the dorsal wall. Okay, it is attached, kidney is attached to the dorsal wall, because of which the peritoneum will cover it only in the ventral region. Now if I draw another organ, now say this is some organ, the peritoneum will completely cover it like this. Okay, okay, but in kidney that is not happening. Okay, clear to all of you? It is attached in the dorsal wall. Now Is this clear? Yes, thank you. Okay, students, so it covers the kidney only by the ventral side. Anyone has doubt in this, please feel free to ask. Okay, I will explain it again. Okay. Yeah, I only prepared the mock test, that's why. Okay, now it is a metanephric kidney. Okay, it is metanephric. Okay, so you know there is pro, meso, meta. Pro will be for your smaller organism, meso will be like for your amphibians, meta will be for higher organisms. So this is metanephric. Metanephric, that means it is made up of metanephrons. Okay, made up of metanephrons. Okay, yes. Clear? Okay, now which you want me to explain again? The metanephric part? I understood. Explain again. Now tell what to explain. Chumma, if you type explain again, what will I explain? Okay, it's clear? Okay, fine. Retroperitoneal means it is basically a cavity. Okay, a space. Retroperitoneal space. So, you people know in the abdomen there is a space. Okay, retroperitoneal space. So, this kidney is present and it is called a retroperitoneal organ. Now, how is the peritoneum covering happening? The peritoneum covering is happening in such a way that the kidney is attached to the dorsal side and the peritoneum covers the kidney only in the ventral side. It is not going to interfere. Okay, okay. Metanephron, students, if you study, it is a type of a nephric tubule. The nephric tubule will have three parts. Okay, pro, meso, meta. Pro will be for smaller organisms, meso will be for the amphibians, and meta, that means will be for the higher organisms. Depth and all you don't have to know, just know metanephric. Metanephric kidney you would have studied many times. Okay, made up of metanephrons. Okay, it is a nephric tubule. Metanephrons will constitute for higher organisms. Clear? If you go in depth, you will not understand a lot of it. So, let us just keep it in the surface level which is required for you. Now, location of the kidney. What is the location of the kidney? It's in the T12, right? T12, okay. T12, yeah, that is 12th thoracic and which lumbar? 12th thoracic to 3rd lumbar region. Very good. Okay, this is general. We are just doing the general. We will get into the structure slowly. Okay, we will get into the structure slowly. Okay, yes, very good. 3rd lumbar region. Okay, yes. Now, we will just see how is this adult kidney. Okay, adult kidney, what is the length, width and everything. Okay, length, width and everything. Okay, now adult kidney. Okay, all of you tell me how much is the length? How much is the length? 10 to 12 centimeter. Length is 10 to 12 centimeter. Width is 5 to 7 centimeter. Correct? 5 to 7 centimeter, agreed? So many things are there, okay? So, sometimes numbers can go wrong. So, please be kind to tell, okay? Thickness, so this is width. Thickness, right? Thickness, yes. What is the thickness? Very good. 2 to 3 centimeter. 2 to 3 centimeter. Yes. Okay. Weight. Last one is weight. Okay. Weight is about 120 to 170 grams. That's all. Okay. 120 to 170 grams. Yes. Okay. Some election community people have come. They're telling vote for some person and all. You people can block. Yeah, if you people spam in my class now, I will make sure to teach you people biology. Okay? If you decided to spam my class, I will make sure to teach you people biology. That is my promise to all of all the students. Okay? Asif, first we will do justice to yourself. Okay, we will do justice to yourself. Do justice to yourself by not wasting your internet, not wasting your time. Okay, and spamming the group. Do justice to yourself. Okay. Then later we'll have to ask justice for Asif. Please give justice, because NEET to you cannot pass by getting justice for big boss people. Correct? Then we'll have to ask justice for Asif because he didn't get into AIMS. Okay. Okay, let's start. Longitudinal side of kidney. Not tired, no? Not tired, right? Thank you, thank you. People will make one reel out of this and send it to me. People make one small snip, ma'am's reply. Okay, let's start. Okay, students, longitudinal side of kidney. Okay, now I want all of you to focus here and draw with me, because this kidney has to look like a kidney for all of us. Okay, it has to look like a kidney, right? So let's try drawing a kidney. Students, this will take a lot of work. Okay, yes. Yes, kidney successful. Okay, now we will draw whatever is inside the kidney. Okay, so students, first I'll tell you few things. This is a kidney. Okay, that is not what I wanted to tell. This is a kidney. Now, what are we talking about the kidney? We are going to talk about the parts. That means what and all is it divided, what and all is there. Okay, that is what we are going to understand. So, understand kidney will be divided into two, cortex and medulla, right? We have discussed this in adrenal gland, right? Cortex, medulla, right? So, ovary we have discussed. So, this cortex and medulla are two regions of the kidney. Okay, so I am going to draw a very thin line to differentiate that. Okay, you people also should draw. Okay, this is the thin line that I have drawn to differentiate. Okay, medulla and your cortex and medulla. Okay, now let's draw some parts. After that, I'll explain. Okay, after that, I'll explain. Now, students, what I've drawn this side. Okay, this yellow color dots is your cortex region. All of you draw with me. This diagram is very important and it will fetch you so many marks for your CBSE, for your NEET. Okay, this dots which I have drawn is the cortex region. Okay, now I have to draw your medullary pyramids. Okay, this is your medullary pyramids. Red is visible to all of you. Colors are visible. Please let me know in the chat box. All the colors are visible to all of you. For me, it is visible, but I just want to know if for you people also it's visible. Yes, okay, great. Okay, so students, I have drawn few things. I will explain you what it is, but before I explain, let's finish the diagram. Yeah, okay. Now, all of you look here. This is a longitudinal side of kidney. Now, how do I say? Okay, now students, this is a bottle. Okay, this cannot be the exact example, but longitudinal side of the kidney is taken. Okay, like this it is taken, not like this. Okay, longitudinal side of the kidney is taken. Now, this is how your kidney will look. First of all, what did I tell you? Your kidney has two parts, right? So, kidney has two regions, that is your cortex and medulla. Cortex and medulla. Okay, some things are there in the cortex, some things are there in the medulla. Okay, longitudinal, that's what I told you, longitudinal transverse section. If this is the kidney, if I cut like this, okay, which you can see from this end, right? If I cut like this, it will be a transverse section. If I take like this, it will be a longitudinal section. Okay, yes. Okay, I cannot with you students. I don't know what you people have drawn instead of kidney in your diagrams that your parents have to interfere. Okay. Your parents' heart will be breaking. This fellow is going to become doctor one day. What will he do with humans? He will he will cut heart instead of kidney, instead of what to tell, instead of hand operation, he'll do leg operation after seeing your diagrams. Okay, so please draw it properly so their hopes increase looking at your diagrams instead of decreasing. Like, no, I'm doing a curse to the country by making this fellow or this girl as the doctor. Okay, so please make sure that your kidney looks like kidney, heart looks like heart, leg and hand, everything looks like whatever it is. Otherwise, your parents will really change the plan, and from today they'll be like, enough of your NEET preparation. Let's do something else. Okay. Okay, fine. Let's start. So, students, kidney has two regions, cortex and medulla. This is clear to all of you, right? It is clear. Now, we have to know what is this parts, what and all I have drawn. Can we label? Can we label together? So, students, if you look here, this is the cortex region. Okay, this is the cortex region. Okay, cortex region. This portion is the medulla. Now, I have to label it everywhere. And this triangular structures which look like a pyramid, right? Yes, I will give you a break. After this, I'll give you a break. Okay. Students, after this triangular structure which is there, that is called as your medullary pyramids. Okay, what is it called? Medullary pyramids. Okay. These medullary pyramids, they start from the cortex and they move. That means, what do I mean by move? That means some portion is in the cortex, some portion is in the medulla, but major portion is in the medulla. These people are called as medullary pyramids. Clear to all of you? Now, this region, right, this region is called your renal pelvis. Renal pelvis, okay. And this green color portion which I have drawn, that is your renal papillae. What do I call it? Renal papillae. Okay. And here we have major calyx, minor calyx, which I will show you in the diagram. Okay. NCRT, no, diagrams will be little here and there, but then cortex and medulla cannot be in the same region. We have already understood that, right? Okay. Yes, got it. So, now students, this space, can you see? This space, can all of you see between the medullary pyramids? That is where your renal columns will be there. So, renal columns of Bertini are present in the between of your medullary pyramids, which is also not mentioned in your NCRT clearly. So, I am labeling it for all of you, so you understand. Okay, this is called your renal column of Bertini. Okay, renal column of Bertini, right? Yeah, column of Bertini is mentioned in your NCRT. Even renal column word is mentioned. So, I'm attaching both the words so you understand where is renal column, where is column of Bertini. Okay, that is basically a space between your medullary pyramids. Okay, clear? Yes. Now, students, renal papillae. Now, this you can see there are small, what to tell, you know, streets. No, how we have small streets, right, between some places. If you have come to Bangalore, if you have gone to Chickpet, you can see that small gullies are there. If you have gone to market, you can see small gullies. Same way, here also in this renal papillae, you can see small pathways which are called as your minor calyx. Minor calyx, okay, and the major ones are called your major calyx. Okay, major calyx. So, minor calyx will open into the major calyx will open into the renal pelvis. That is the pathway. Okay, that is the pathway. So, that pathway I will teach you when I teach you blood flow, but just understand this portion, because in your textbook only calyces are given, papillae is not given. So, that is why I am mentioning you. Renal papillae means they will have this small portion called as renal calyx, minor calyx, and this larger portion as the major calyx. Both of them will lead to renal pelvis. Clear? Bertini's position is here, dear. Between the medulla, between the medullary pyramids, the space that you see, no, that is your renal columns or columns of Bertini. Okay. Now students, one more thing, after that I will give you a break. Okay. Now this kidney has to be protected. Agreed? Kidney has to be protected. Chumma will leave the kidney like that. No, no, we have to protect it. Okay. So for the protection, there is an envelope. Now that envelope is made up of three things. Okay. That envelope has three things. Okay. Now this line that I am drawing, green color line that I am drawing is called your renal capsule. Renal capsule. Okay. On the top of the renal capsule, there is going to be this circular structures adipose capsule. Adipose capsule. And top of that you will have like this bracket kind of structure which is called your renal fascia. Okay. Renal, I'll write it, I don't know where I'll write it. So in this one, okay, renal fascia, okay, they are, okay, adipose capsule, okay, red you cannot see, no, one second, I'll erase red color, okay, adipose capsule, adipose capsule, renal fascia. They are together. Students, why is adipose needed? Adipose will do the shock absorber, like whenever we are moving, whenever we are exercising, kidney has to be safe, right? So for that, this adipose layer will make sure that it absorbs all the shock, okay? This three together, who and all, I'll write it this side for you people because now here the diagram has gone really big. So students, here renal capsule, okay, renal capsule, down renal capsule, okay, above that we have adipose capsule, above that you have renal fascia. This together, this all three together is called the envelope that protects the kidney. Okay, this will be present throughout the kidney, but I have only drawn it one place. Okay, to understand all of you, it is just a protection, students, don't break your head. It is just an envelope to protect the kidney, which is made up of three layers. Okay, three layers. It's not major pelvis, it's called major calyx. Okay, renal pelvis. This minor calyx will open into the, oh yeah, I wrote it wrong. Sorry. Renal calyx. Thank you. Who is that? Srinath. Thank you. Major calyx, which will open into your renal pelvis. I made khichdi of both the words. So I wrote easy way, major pelvis. Renal papillae is basically a combination which has your papillae means what? Those structures, right? Which will increase the, which will be, what you tell, like how you have villi, microvilli, like that. This will also be like a little coil structure which will increase a little surface area. So renal papillae together, that means it will have the small street like or gully like portion which is called the minor calyx, that will open into the major calyx, and that major calyx ultimately will open into this portion called as the renal pelvis. Okay, that I'll teach you in the, when I'm talking about blood, flow of blood. Now you people just understand the labeling. Clear? Okay. Yes, we will complete. Clear? Nandini, clear? Okay, fine. So students, is this clear? Is this envelope clear? It is basically a protection for the kidney. Okay, it will do what? Whoosh. Sorry. The sound came out naturally, I'm sorry. Okay, it will basically do protection for kidney, and it is made up of three layers. Okay, it is made up of three layers. Okay, got it? So kidney is divided into cortex and medulla. Clear? Cortex will be the outside part, medulla will be the inside part. Medulla will have a form of pyramids called medullary pyramids. Okay, some part of the will be in the part of the cortex, some will enter the medulla. Okay, and renal papillae have small pathways which are called as minor calyx that open into the major calyx and then open into the renal pelvis. Clear to all of you? This is all about your longitudinal portion. Now, last thing, I think 2 more minutes. I'll give you break, okay. One more thing, one more last thing to do is your hilum. This is your hilum. Have you seen in your Rajma also there is a small portion like a scar like portion which is called the hilum. Here it is a notch like structure, right? It is a notch like structure which is called your hilum. It is a notch like structure. Okay, notch like structure. Now what and all hilum will do? Okay, hilum, through hilum what and all is going to pass? Your ureter is going to pass, blood vessels are going to pass, nerves are going to pass, right? From with hilum, so many things are going to pass. Okay. Students, I have written here hilum, through hilum, ureter is going to pass, blood vessels are going to pass and nerves are going to pass. Okay. Got it? Clear? Oh no, quiz came off. Okay, let's take a break. What's the time now? 6:34. Okay. Five minutes break is enough? 5 minutes break? Yes? I don't want to waste your time. Tell quickly. 5 minutes is enough. No, only 1.5 hours got over. Okay? Yeah, 5 minutes is enough. All of you come back at this time. Okay? Without fail. Are you all back? Yes, 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 yes. All of you are back. Let's see the number. Students, all of them are not back. Quickly text them. Yes. I'll also text. Hello, students. I'm back. Are you all back? Yeah, I'll reply to your meme when we reach the last of the chapter. When I talk about dialysis, I'll teach you this. Okay? Okay? Yes, students are not back. Yeah, so many went off. See, this is why I should not give break. If you give break, no, they'll go off. They'll go sleep. Yeah, this box won't go. Students, 1 minute. Yes. Okay. How is the Josh? How is the Josh? Hi. Yes, all of you are back. Yeah, number is not improving. Around 50 students are missing. Students, everything is depending on NCRT. Every single line is NCRT, okay? Do not confuse your friends and confuse yourself. Okay, do not confuse your friends and confuse yourself. Let everyone join back quickly and we are going to start. By then, students, revise. You have a quiz coming. You have a quiz coming. Okay? Yes. Okay. Ready? 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 Okay, here goes your first quiz. So, I am going to draw lines. You have to label those parts. Are we ready? Yes? Gopika ma'am is here. You can't see? Yes, osmoregulators have all come. Osmoconformers have gone somewhere. Right? Okay, I am going to draw the parts. You are going to label. Okay? All of you can keep a score of yourself, and when the session ends, you can comment down your marks. Okay? You can comment down your marks. Okay? Yeah, no more breaks till dinner. As Sai wants to kill me. Okay, that is part 1. Part 2. Okay, all of you write 1, 2, 3 and label these parts and let me know. After that, I will give you the answers. After that, I will give you the answers. Okay, you have to go like 1, 2, 3. Okay, 1, 2, 3. Quickly. Yes, very good, very good. Very good. Medullary, cortex, pelvis. Okay. So most of you have got all the 3 marks, right? Anyone who has made mistakes? I don't think anyone has made mistakes. So shall we write the answer, or do you want some more time to type? Yes. Medullary pyramid. Okay. What's the second portion? Renal pelvis. Okay. And this is cortex. So all of you have scored 3 out of 3. So your first score is 3. Okay. 3. All of you scored 3. Yes, 3 marks. Good job. Okay. Cortex. This is cortex. The arrow mark is not visible. This one. Okay. This one. Cortex. Okay. Okay. One quiz is done. So at one quiz you have scored 3 marks. Keep a count of your scores. I cannot keep count of so many students, right? Yes. Okay. 3 is confusing. Why is it confusing? Look here. This portion, wherever your medulla is there, right, your medullary pyramid is little towards the cortex and more towards the medulla. So wherever it is starting, that means it is cortex. Clear? And students, all of you look here. Can you see a nephron? Can you see a nephron? So if I am drawing this, right, if I am going to my diagram and if I am drawing, I will draw a nephron here. I will draw a nephron here. Can you see a nephron that I have drawn? Can you see a yellow color nephron I have drawn? Can you see? Did I label it wrong? This is renal pelvis, this is cortex, this is medullary pyramid. Correct or no? So students, here is the nephron in the medullary pyramid. Okay, clear? Yes. Now ureter. Okay. Yes, ma'am. 3 is renal pelvis. Yes, got. Oh, 3 is not visible, you people are telling. This portion. Okay, this portion is your renal pelvis. This is your ureter extending downwards. Clear? Okay, fine. Students, ready? Next, we are going to study about your ureter. Now, all of you know about your ureter? Yes or no? All of you know about your ureter? It's like this, right? It's like this, your ureter. Now, don't you think when urine is coming from the kidney after purification, when it is coming, the ureter will pass it to your urinary bladder. Your ureter will have to do little kind of like a little stretching, little improvement in the dimension, right? It has to increase a little so that more urine can pass. All this has to do, correct? For that, there is a set of some muscles. There is some set of muscles. So, they have a 3-layered, okay, 3-layered wall that helps them for stretching, that helps them with increasing the dimension. Clear? Now, we are going to study about that 3-layered wall. Okay, now we are going to study about that. No. Okay. Now, see here. Let's draw it in one color. So, first one, I'll draw it thicker. This layer, okay, is your transitional layer. What is this layer called? A transitional layer. So, what are we talking about? We are talking about 3 muscular layers which will do the job of stretching, increasing the dimension so the urine can pass to the urinary bladder, right? So, that is your transitional layer. So, this first layer is your transitional layer. Clear to all of you? Then, the second layer, let's draw it with orange, okay. This layer, okay. Oh, is called your muscular layer. Okay, muscular layer. This is, I am talking about your ureter. I am talking about your ureter, right? First layer is your transitional epithelium or your transitional layer. Okay, so we will write here transitional epithelium. That is the most appropriate word. What will it help for? It will help for stretching. It will help for stretching. Okay. And then we have a second layer, which is your muscular layer, that is again going to help in increasing the dimension. Okay. Okay, got it? Longitudinally, dear. Okay. Last layer is going to be a connective layer. Last layer is going to be a connective layer. Okay, a connective tissue layer. All of you got the 3 layers? All of you got the 3 layers? Yes or no? So now what is this 3 layers going to do? They are going to help for peristalsis. That means contraction, relaxation, contraction, relaxation. Okay, it is going to help for peristaltic movement. Peristaltic movement. Clear? Yes or no? This is your ureter. Students, remember the color. Blue color is going to be transitional epithelium. Orange color is going to be muscular layer. Green color is going to be connective tissue, because I am going to show you for urinary bladder. Okay? Clear? Can we go to the next one? Now, both the ureter will open into the urinary bladder. So, now we have to study urinary bladder. Okay? So, this is your urinary bladder. Agreed, all of you? Now, let's start with the first layer. What will be the first layer? Oh, students, I did some magic which I am not sure about. Urinary bladder got cut into 2 pieces. Okay. What is the blue color layer? What is the blue color layer? Epithelium, endothelium. Endo means inside, epi means outside. Correct? Jyoti. Endothelium means inside, epithelium means outside. Correct? Here also what are we seeing? It's endothelium is inside, epithelium is outside, right? Yes, transitional epithelium. Very good. Transitional epithelium. Okay. Then we will have what? Muscular layer, right? Okay. We will have a muscular layer. Students, in your NCRT, it is given smooth muscle layer. Okay. It is given smooth muscle layer. Okay. Okay. Spam people have come. I hope all of you block and continue. One second. Yes. Okay. So, students, second layer is your muscular layer. Okay. Now, in muscular layer, you have to know the muscle is detrusor muscle. Now, why this muscle is important? Because this muscle is going to help in contraction. When it contracts, it will allow the urine to pass out, right? So, it will help in contraction of your urinary bladder. Causing urine to pass out through the urethra. Okay. So, in your textbook, it is given smooth muscle. Okay. It is not detrusor muscle is not mentioned in your NCRT, but it is okay for you to know detrusor muscle is the smooth muscle which helps in contraction of the urinary bladder. So, the urine passes out. Clear? Okay. Now, the last layer is what? connective tissue, right? Connective tissue. Clear to all of you? Okay, we have what and all? We have what and all? Students, what happened to the Josh? All of you slept off. Quickly hit the like button and also show me the Josh. Are you all there? I cannot see the chats moving. I think it's a problem with my chats. 1 second. One second, students. Now it's loading. I am able to see all your love. Wow, that's amazing. Yes. Spammers, if you're coming, please like the session and go. Anyway, you wasted your energy to come here and type a message. So, in that time, take 1 minute to like the session. Okay. It will be a lot of help to the community. Okay. So, spammers, don't worry. Come, like the session or fir go. Okay. Yes. Okay. Yes. No times to look at the chats. Exactly. Okay, students, look here. So, what I was trying to say, connective tissue is there, muscular tissue is there, which is called the detrusor muscle. It is going to help in contraction. It is going to help in contraction. Now, imagine this is your urethral opening. Okay, this is your urethral opening. Right, this is your urethral opening. Now, your urinary bladder forms a structure, a triangular structure. Before that, let's also draw your ureter coming into the urinary bladder. Ureter is coming into the urinary bladder. Agreed, all of you? Now, this will form a triangular structure which is called the trigone of urinary bladder. What did I say? It is called the trigone of the bladder. Okay, this triangular structure which is formed with two urethral opening and two ureter opening and one urethral opening is called your trigone of bladder. Okay, trigone of bladder. Clear? Now, why is it called trigone? Why is it called trigone? Okay, it is called trigone because there is two ureter opening, right? Why is it called trigone? Because there is two ureter opening and plus one urethral opening. Okay, so that is why it is called trigone. Okay, clear to all of you? Clear to all of you? Trigone is because of two ureter opening with one urethral opening. Okay, urethral opening. Okay, yes. Now this is clear. Okay, this is clear. Let's understand our sphincter muscles. Okay, sphincter muscles. Okay, we have two sphincter muscles. Okay, we have two sphincter muscles, sphincter muscle 1 and sphincter muscle 2. Now tell me which is going to be involuntary, which is going to be voluntary. Which is going to be involuntary, which is going to be voluntary. All of you. Which is going to be involuntary muscle? Is this sphincter muscle 1 or sphincter muscle 2? Is it internal sphincter? So, can I call this internal? I can call it internal. I can call this external. Right? Internal and external. Very good, very good. So, internal one is involuntary, right? In for involuntary. Okay. So, the internal sphincter is involuntary. That means it is not in your control. You cannot control when it should open, when it should relax, when it should contract. That you cannot do. Okay. That you cannot do. 1 minute. Why? Okay. So, internal spinster is involuntary, external spinster. Voluntary. Clear to all of you? External is voluntary. Now, how much urine can your urinary bladder hold? How much urine can your urinary bladder hold? Okay. How much urine can your urinary bladder hold? Darshan Somashekhar, thank you so much. Thank you so much for your knowledge. Okay. If I want to refer encyclopedia, I will definitely contact you. Now, please stop and go. Okay. Do not forget to like the session. Yes. Okay. Yes, yes. How much? 700, 800. Okay. I like the dedication he has. He's an osmoregulator, students. How much it can hold? 700 to 800 ml. Yes, it can hold up to 700 to 800 ml. So, what will happen? Now, the spinster muscle, one is involuntary. That's when you will ask your teacher that, ma'am, may I use the washroom? Maths teacher will tell, no, no, you cannot use the washroom. Go sit there. Go do the problem. That time what is going to happen? Your urinary bladder will be 500 ml full. Again you will sit, you will be like, okay, I'll do one more exercise. You will check the time for the bell to ring. Okay, then it will become 600, 700. By the time it reaches 700, 750, your spinster muscles also will be like, okay, I need to relax now because I need to void the urine out. So that is when you will be like, ma'am, please, may I go, ma'am, please. That is how your tone also will change, right? Right? Your tone also will change. Yeah, it is in NCRT only. How many times you people will ask? Everything is in NCRT. Tell me one thing that is not there in NCRT. Spinster muscles you have to know because they are asking you how does voiding of urine happens. That means you have to tell contraction of urinary bladder and relaxation of your spinster muscles. Correct? Okay. Now students, what is happening? Urinary bladder will contract. Okay. Spinster muscle will have to relax. If it contracts, it is not going to leave the space. Got it all of you? Now, if it contracts, it is not going to allow the urine to come out. But if it relaxes, it is going to allow the urine to come out. Got it? Got it? So, when urine has to come out, what are the information that you need to know? The information that you need to know is that urinary bladder will contract, okay, and your spinster muscles will relax. Got it all of you? If, okay, if urinary bladder contracts and spinster muscle relax, then only urine will be able to pass out. Clear to all of you? That is why it is important for you to know your involuntary and your external and internal spinsters. Is this clear? Is this clear? Now what is going to happen, students? Now this is your urinary bladder, right? It will have something called as your stretch receptors. It will have what? It will have stretch receptors. Imagine it has stretch receptors. Okay, now the stretch receptors will get an information that the urinary bladder is full. Okay, urinary bladder is full. It will send message to the brain. So, brain will send message back what? Telling that void. Micturition reflex will happen. That means let micturition happen. That means let the urine be expelled out. But does brain know that you are sitting in a maths class? No. Does your stretch receptor know that you are sitting in a maths class? No. What do they know? They know that when time is up, when there is 700 to 800 ml, urinary bladder knows that it has to contract. Okay? It has to contract. Clear? Clear to all of you? Yes, no? Yes, 1 second students. I will just beautifully throw the spammer out. Give me 1 minute. By then write all this what I have written on the board. Okay. Where is it? Where is 1 second? Yes. Are we done, students? Did you all finish writing? Yes. Okay. Okay. Yes, fine. So, now students, there is one condition. Now, urinary bladder contracted, your sphincter muscles, right? Everything has happened, okay? Now, what is happening is, if a person, okay, if a person is not able to understand that the urinary bladder is getting full, that means what is happening? If you are happening to not know that the urinary bladder is getting full, okay, what is going to happen? Ultimately, urine is going to leak. That kind of a condition is called as incontinence, okay, incontinence, okay. That kind of a condition is called as incontinence. That means when your urinary bladder completely gets full, okay, completely gets full, and your stretch receptor is not able to send signal to the brain, urine will leak, right? That condition is called incontinence. Incontinence. Okay, clear? Cleared all of you? Cleared all of you? Yeah, too many spammers has come. I think after 6 o'clock, 7 o'clock is the time for spammers to come. Yes, who and all? Yes, done students. The job is done. Okay, so students, yes, look here. Incontinence, okay, incontinence means what? When the urine leaks and when they do not know that the urinary bladder is getting full. Clear? Okay. Next, what we are going to do is your urethra. What are we going to do? Urethra. So, students, urethra, what is urethra? It is basically helping the bladder to void out the urine. That is, from the bladder, the urine has to go out, right? That job will be done by who? Urethra, right? Urethra. Now, in male and female, the urethra is going to be different. Male and female urethra is going to be different, okay? In male, this urethra is going to be longer. Is going to be longer. So, they have longer urethra, okay? And because this in male, the sperm and urine will be passed by the urethra. So, we can call it a urinogenital duct. Can I call it like that? Yes, I can call it like that. Urinogenital duct. I can call it like that. Definitely, I can call it like that. That is because it is a common passage. It is a common passage for urine and sperm. Urine and sperm. Okay, clear? Okay, now you people tell me, this is longer urethra, urogenital duct, common passage of urine and sperm. Urine and sperm will not come out at the same time because urine's pH will kill the sperms. Clear? Now, when we talk about females, okay, let's talk about females. Students, females, the urethra is short. They have shorter urethra and it is not a common passage, only urine will be passed out. Okay, only urine will be passed out. Okay, only urine will be passed out. Clear? Okay, clear? Only urine will be passed out in urethra and the urethra is short. Now you people tell me, among male and female, who has high chance for UTI, urinary tract infection? Who has a high chance? Yes, female affected by infection. Karuna, very good. Females have high chance for UTI. Why, students? Because the urethra is short, it is easy for the infection to pass. Correct? Very good. Females. Okay. It is easy for the infection to pass. Cleared all of you? Cleared all of you? Yes. Did you all like the session? Hitman need a hit. Cockroach hit. Okay. Yes. Done, done. Are we done? Yeah, you go ask in sky, please. Mintu Deep, go to sky, please. Go ask elsewhere. Why are you asking here? Okay, done. Let's do nephrons. Okay, students, I thought I can draw it in front of you, but I realized it's going to be really difficult because it's not very easy to draw on board. Okay, so I have drawn it. Now all of you please draw it. Okay, all of you please draw it. UTI is urinary tract infection. Have you felt that very bad abdominal pain? You'll feel irritation. Okay, you'll feel irritation that you'll feel like the doctor will tell you drink a lot of water, drink barley water and all that. That is because you're in the urethra, like from the urethra, the bacteria has entered. Okay, got it? Yes, fine. yeah, I have to drink water. Okay, students, now let's talk about nephron. Students, nephron is the structural and functional unit of the kidney, correct? Nephron is the structural and functional unit of the kidney. What am I trying to say? Nephron is the hero. Whoever we were talking about, they are all side actors. Okay, urinary bladder, urethra, ureter, kidney, all those people are side characters. Who is the hero? Hero is nephron. I told you, picture abhi bhi baki hai because we are getting into the picture. Right? We are getting into the picture. Got it? Students, don't shoot into the gun. Let me teach. Don't ask questions that I have to teach you in the end of the chapter. Okay? Yes. So, students, look here. This is the general structure of a nephron. So, first what I have to write? It is a structural and functional unit of kidney. The real hero, the real hero, right? Nephron is the hero. All other people were side actors, but we were giving so much importance to it, right? Okay. So, students, this is a nephron. Let's start labeling the nephron. Okay. Now, nephron, when I talk about nephron, I can split nephron in mainly into two. Okay. I can tell nephron has two main parts. Okay. One is called the renal tubules. And one is called glomeruli or your glomerulus. Okay, glomeruli or your glomerulus. Now, when I talk about renal tubule, whom am I talking about? I am talking about your PCT, proximal convoluted tubule, loop of Henle and DCT. Whom am I talking about? I'll write all this full form in the next slide. Do not worry, just write this. Okay, PCT, loop of Henle and DCT, proximal convoluted tubule, loop of Henle and DCT. These are the parts of the renal tubule. Glomerulus is having, it will be present inside the Bowman's capsule. Now, this is the Bowman's capsule. So, shall we start labeling? This is Bowman's capsule. What is this red color portion that I have drawn? What is this red color portion? Let's draw some. This is glomerulus This is glomerulus. Yes, all of you are with me? Yes. Bowman's capsule, glomerulus. This portion, students, it is very convoluted. It is very like you tell, it is more tubes are present. But if I draw like that, I will never be able to explain it to you well. So I have drawn a more separated diagram. Okay, this portion is your PCT. Okay, this portion is your PCT, proximal convoluted tubule. Okay, got it, all of you? This is your PCT, not the red color, this white color. Okay, red color is your blood vessels which are passing. Now, this portion, which is the U-shaped portion, this U-shaped portion is called your loop of Henle, but loop of Henle has two portions. One is called the descending lymph, okay. This is ascending lymph. Okay, what did I tell? Descending lymph and ascending lymph. Descending lymph which is coming down, ascending lymph which is going up. And this U-shaped structure is called your loop of Henle. It is like a hairpin structure, right? We have studied this in our smaller classes. We have studied this loop of Henle, hairpin like structure, U-shaped structure. Yes, very good. Okay. Then when we go here, we will see our third portion, which is your DCT or your distal convoluted tubule. And then which you see this long tree like structure which you are seeing here, this is your collecting duct. Collecting duct. Clear? Clear to all of you? Collecting duct. Okay. Now I will tell you about the what are the blood vessels passing and all that. So let's label that in orange. This one which is coming in is called your afferent arterioles. Afferent arterioles, which is going out of the glomerulus, right? Which is going out is called your efferent. Okay, it's called your efferent arterioles. Got it? Efferent going out. Okay, afferent coming in. Okay, afferent and efferent. Okay, clear to all of you? Then this network like structures which you are seeing is called your vasa recta. They are also a type of capillaries. Okay, type of capillaries called vasa recta. And this portion which you are seeing here is called your peritubular capillaries. Clear to all of you? All the labelings clear? Yes? Yes, vasa recta, peritubular capillaries, afferent arterioles, efferent arterioles. Yes. Got it? So, who is bringing the blood in? Afferent. Who is taking the blood? Efferent. Okay. I hope all of you have drew the diagram as well. Did you all draw the diagram as well? Okay. Yes. Okay. Now, we will write these information in the next page. Okay. We will write all this detailedly in the next page. So, I am just erasing this. So, this page has only the diagram. This is for your notes purpose. That's why I am maintaining it. Okay. Yes. Let's write everything about it, students. So, first we will write about our nephron. What is our nephron? Structural and functional unit of kidney. Okay, structural and functional unit of kidney. Correct? Now, I can divide nephron into two parts, right? I can divide nephron into two parts. That is renal tubules. I'll do one thing, I'll write here glomerulus. Glomerulus, renal tubules. right? Now, renal tubules are of three types, right? Proximal, convoluted tubules, okay, loop of Henle, distal convoluted tubules. Correct? Agreed all of you? Okay. Done. Now, this is how much we know about nephron, right? This is how much we know about nephron. Now, we know afferent will bring in, efferent will take out, right? And then we also know peritubular capillaries and your vasa recta, okay? Okay. No, nephron, see look here, I'll tell you who has asked the doubt. Dr. Strange. See this, if I'll draw it here. If I say this is your medullary pyramids, okay, medullary pyramids are here, okay, this is where your nephrons will be there. This is how your nephrons will be placed. Can you see? This is how your nephrons will be placed. Clear? Got it? This is how your nephrons will be placed. Is it clear, Dr. Strange? Yes. So, here you can see some portion is to the medulla, some portion is outside, right, to the cortex. Clear? Okay. Now, students, you cannot call it urine. You cannot call it urine until it is in the collecting duct. So, what will you call it throughout the process? Whenever it is in PCT, loop of Henle, DCT, everything you are going to call it a filtrate. Even when it is in the glomerulus, you are going to call it a filtrate. You will call it urine only when it is in the collecting duct. 1 4-mark question. Okay. If you have listened to this class, maybe this question comes, you will get 4 marks. Clear? Clear to all of you? It's not wound around, it is just placed like this. Can you see? Some portion is in the medullary, some portion is outside towards the cortex. You cannot tell it is wound around, it is just a placement like that. Okay? Okay? Got it? You cannot call it urine until it is in the collecting duct. So, the question can be, when is it called urine, or if can it be called urine if it is in the PCT? You will call it urine only if it is in the collecting duct, otherwise you will call it filtrate. Okay? Filtrate. Clear? Fine. Now, students, we have to study types of nephron. What do we have to study? Types of nephron. Now, one more question can come. Is collecting duct part of the nephron? Is collecting duct a part of the nephron? Yes or no? Yes or no? Is collecting duct a part of the nephron? Yes, no, okay. Maybe, maybe. Come on, students, maybe and all you can't tell. It's neither yes or no. Okay, the answer is no. The answer is no. Collecting duct is not a part of the nephron. Why? Because many nephrons open into the collecting duct. Many nephrons open into the collecting duct. This is a nephron opening into the collecting duct. This is a nephron opening into the collecting duct. This is a nephron opening into the collecting duct. Because of which multiple nephrons open into one collecting duct. Because of which we cannot tell collecting duct is a part of the nephron. Clear ho gaya? Yes, no? Clear? Okay. Now, students, we have to understand types of nephron. Okay. Let me see. Oh no, I haven't left slide. So, in 1 second, we will just add one slide. Okay. So students, we have to study types of nephron. Now why do I have to study types of nephron? Types of nephron is depending on how much the loop of Henle is going into the medulla. What did I say? Loop of Henle, depending on how much loop of Henle is going into the medulla, it is divided. The nephrons are divided into two types. Clear to all of you? Clear to all of you? Okay. It is called cortical nephron and juxtamedullary nephron. Juxtamedullary nephron. Okay. Yes. So students, your loop of Henle, depending on how much it is going, it will be divided into cortical and juxtamedullary. Okay. Now let's discuss few things about cortical. Students, first I will draw a diagram. You can look at the diagram and you can tell me, ma'am, like if you understood or not. Okay, if you understood or not. Now think this is the border. Okay, here you have cortex, here you have medulla. Okay, now I am going to draw one glomerulus. I mean, I am going to draw one whole nephron. Okay, so you can tell me if you are understanding. These are two nephrons. Okay, what I have drawn this is two nephrons. I have not drawn in detail or anything. I have just drawn to show you. Okay, I have just drawn to show you. Okay, can you all see these two nephrons? Yes. Okay, first one is your cortex. Okay, cortical nephron. Okay, what is wrong, Asif? First one is cortical nephron. What is cortical nephron meaning? Cortical nephron meaning it will touch, the loop of Henle will briefly touch the medulla. Loop of Henle will hardly touch the medulla. Okay, clear? Got it? So, when I write the point, what am I going to write? Loop of Henle will briefly touch the medulla and 85% is cortical nephrons. In our body or in our kidney, 85% is cortical nephron. Okay, clear to all of you? 85% is cortical nephron. Loop of Henle will briefly touch the medulla, that is what you are seeing here. And I have drawn the blood vessel. If you see, I have drawn the blood vessels in such a way that it is not touching the loop of Henle. That means vasa recta is absent or vasa recta is not there in your cortical nephron. Okay. Vasa recta is absent in your cortical nephron. Clear? Clear? Yes or no? Yes or no? Now, when do these cortical nephrons work? Cortical nephrons mainly work when they are in normal condition. Normal condition means now your and my condition. That means we both are drinking water, we are using the washroom, we are doing everything. Thank you, Dr. Strange. Okay, 85% is your normal cortical nephron, that means normal condition. When you are drinking enough water, when excretion is happening, everything is proper. But if a person is fasting, what will happen? That time juxtamedullary nephron will do the job. Juxtamedullary nephron will do a lot of help. Okay, so here we have 15% of nephrons are juxtamedullary, and this will mainly work when it is in stress condition. It is mainly work when it is in stress condition, and loop of Henle will be deeply in medulla. Clear to all of you? Clear to all of you? Yes or no? Clear? Students, 2 minutes, I'll just sit. By then, please make a note of this. Okay? Yes. Okay. Juxtamedullary is less in number because they are mainly used only in stress condition. Since you already know that DCT and collecting duct will do maximum reabsorption or conditional reabsorption when situation is unfavorable, right? So, when they are more into the medulla, this kind of, when the loop of Henle is longer, that means we will get more of reabsorption. That means when the condition is really stressful, we will require more reabsorption. Okay? Yes, yes, right. Should I stand this side? Yeah, right. All of you. Yeah, yeah, I've moved. Okay. Hmm. That's what I just now told Siddharth. It will help in maximum reabsorption. It will help in maximum reabsorption. Cortical, what is it, nephron works in normal condition. That does not mean it is not working in when a person is fasting. When a person is fasting, juxtamedullary will get hyperactivated or it will become more active so they can reabsorb as much water so the person does not lose any water and will be able to actively be okay, right? There are a lot of people who thank you, Karuna. There are a lot of people who fast, right? Who stays without even, taking in any saliva. So, how do they survive? Their kidney also has to do excretion. For urine, we need water. So, that time, this kind of nephrons will do extra job. Clear to all of you? Okay. Vasa recta, vasa recta. Here they have vasa recta. If you look here, they have vasa recta here. Okay, vasa recta is present. Clear? Yes? Okay. Okay. Now, next topic is how does this blood flow? How does this blood flow? Students, now we are doing the general, okay? We are doing general, okay? We are not doing, we will do PCT, then we will do DCT, then we will do loop of Henle, then we will do urine formation. Then in your brain, one khichdi will be formed. So, what we are doing is, I am teaching you general. I am teaching you just this is what nephron is there, this is what is happening. Now, I will teach you how is this blood flowing for purification, okay? How is this blood flowing for purification? Once the blood flow for purification, then if I teach you urine formation, it will make more sense. Clear? Okay, clear? Okay, let's do flow of blood. So, now blood is entering the kidney. All of you know that blood is carrying urea which has to get purified and it has to go back. Okay, so we will start from iota, right? We will start from iota. Did you all like the session? Did you all mark your attendance? Whoever joined late to the class or latecomers, backbenchers. Okay. But Aishwarya, from head you cannot take a NEET. From head you cannot take a NEET, no? Then later you will go see the question paper and tell, oh no, Gopika ma'am taught us in this order, question came in this order. Okay, so let's be careful. Did you all mark your attendance? Let me see the number. Students, I told you I am very strict. You cannot bunk my class. You have to mark your attendance. Okay, before I check quickly, like, this much love is enough for my effort. Yes or no? I'm even cracking jokes so you people don't sleep. This much love is enough. Yes or no? Not enough, not enough. Quickly, like all the latecomers. I will take attendance every 1 hour. So you cannot escape also. Yes, okay. Students, no bunking. They are capillaries. Vasa recta are capillaries, type of peritubular capillaries. Okay, they all arise from the efferent arterioles. Varalakshmi, I told 2 p.m. class, but all of us are going in uniform. 5 p.m. is, most of the students are coming back from college. They also need a break. So I thought I'll take it at 5 p.m. Okay. Yes, fine. So students, we'll start from aorta. What did I tell you? Is our artery carrying oxygenated blood? Yes, it is carrying oxygenated blood. But is it toxic free? Is it toxic free? Yes or no? Is it toxic free? I told this in the starting of the class. Very good, very good. It is not toxic free. It has toxic. What is the toxic substance it has? Urea. Right? It has urea. That blood is coming to kidney for purification. So, aorta, right? From aorta, it is going to come to your renal artery. From renal artery, where will it go? From renal artery, it will get into your afferent arterioles. It will get into your afferent arterioles. From afferent arterioles, where will it go? Can any of you guess after afferent arterioles? Urea is toxic. No, we cannot keep urea. So, we are going for purification, right? So, it will go to glomerulus. From glomerulus, it will go to all your renal tubules. But after that, afferent arterioles, correct? From afferent arterioles, peritubular. Peritubular capillaries, correct? And then finally it will get purified and it will go out with the help of renal vein. Renal vein, which will have deoxygenated blood. That means carbon dioxide will be present, but it will be purified. That means it will be having no urea. Clear to all of you? Blood flow? Blood flow is clear. Students, your renal tubules will not come. By mistake, I told renal tubules will not come, because what are we talking about? We are talking about blood flow. We are not talking about urine formation, right? So, look here. So, from where is this blood going to come? I think this one. Students, blood is going to come from where? It is going to come from your afferent arterioles, right? From which color do I do? Say blue I do. So, students, it's coming from your afferent arterioles, it is going to your glomerulus. From glomerulus, it's going to afferent. Afferent, say it is going down. It will go down to vasa recta, to peritubular. From peritubular, it will come, right? It will move to your renal vein. Clear to all of you? Clear to all of you? Yes, look at my arrow marks. So beautiful. They're dancing. Can you all see? Yes. Okay, great, great. Thank you so much, Jasmine, Mishra. Okay, so yes. So, flow, flow, students, flow. My flow is gone. My tongue is twisting. Okay, flow of blood is aorta, renal artery, afferent arterioles, glomerulus, afferent arterioles, peritubular capillaries, renal vein. Renal vein will have deoxygenated blood, but that blood will have carbon dioxide and that will have no urea. Clear? Yes, yes. Vanakkam, Vanakkam, Vignesh. I know Tamil. Students, telegram. Oh no, I don't know why I always write this. Biology by Gopika, okay, telegram channel, biology by Gopika, you can join, my DP is put, okay, yes, clear, fine, done. Now we are going to one amazing, like this is love of my life, this topic. I'll tell you why it is love of my life, because once you understand this, no, It's going to be so beautiful. That's why it's love of my life. Now, this is a process. Now, all of you kept asking me, ma'am, how does this work in fasting? How does this work in fasting? How does it work when the conditions are, going here and there, right? Students, there is some apparatus called as Juxtaglomerular apparatus, or your JGA. Juxtaglomerular apparatus, or your JGA. Now, what is this JGA going to do? When the conditions are really, really bad, okay, when the conditions are really, really bad. Yes, one second, yeah. When the conditions are really bad, what do I mean by conditions are really bad? There could be chances that your BP is low, H2O is low, okay, in the blood, okay, or else volume of the blood volume is low. In these conditions, there should be someone to help, correct? Your sodium level is low, your water is low. When these conditions happen, there should be someone to help, correct? Correct? Okay. That time we have an apparatus called as JGA apparatus. Now, I have drawn it upside down. Now, what is this cup-like structure? All of you tell me, what is this cup-like structure? Tell me quickly. What is this cup-like structure that I have drawn ulta? Quickly. Gayatri, session will end when I finish teaching. I seriously don't know when the session will end. We have again urine formation. Maybe it will end in another 1.5 hours, 2 hours. I don't know. Okay, so students, yes, very good. This is Bowman's capsule. Bowman's capsule. Correct? Okay, this is drawn ulta. Now, what is this portion? This portion, which is looking like a Cheetos packet. Orange and yellow is Cheetos packet, no students? I hope I'm not very old generation also that I'm telling wrong. Right, this is glomerulus. Very good. This is glomerulus. Okay, this portion is glomerulus. Now let's get into what is happening. Okay, students, this portion, this entering, right? What is entering the glomerulus? Which is that? Afferent arterioles. Afferent arterioles is entering, right? Who is exiting? Who is moving out? Afferent. Okay, afferent arterioles. Correct. Very good. Okay. Dr. Stranger, this is the only way to explain this. There is no other way to explain this. If I draw it like this also, you cannot understand. This is the exact way to draw because I have to show you this also. Okay, students, this portion, this circular portion which I have drawn is none other than your DCT. DCT. Okay, students, look here. Look here. See, when I drew this here, I drew DCT and glomerulus very close to each other. Not very, very close because I had to label, but otherwise I drew it very, very close. That means this is your Bowman's capsule. This is how your DCT will be. Okay, got it? That means DCT and your glomerulus, your Bowman's capsule are in very close proximity. But in diagrams, we do not draw it that way because it will confuse the student. Got it? Like how Dr. Stranger was telling, it will confuse. Okay, so that is why they draw it with lot of space, but it is actually very close to each other. Okay, now look, keeping this in your head, I want all of you to look here. Students, look here. Afferent arterioles are there, afferent is there, Bowman's capsule. This is a portion of your DCT. This is a portion of your DCT. That means if I take a DCT, if I take a tube, okay, if I take the DCT like this and I cut one portion, right, I cut one portion, I'll get one circular portion, agreed? I'll get a circular portion, that will be your DCT. I haven't started explaining, dear. I am just labeling this. I haven't started explaining. Calm down. Okay, look here all of you. This is your Bowman's capsule. DCT is very close to it. Now, I've taken this much portion. and I've tilted it and drew. Okay. So, Now look here, this is an apparatus that is going to help when your BP is low, water is low, blood volume is low, or when it is high, when your conditions are going here and there. Okay, so I have taken DCT, this is your DCT. I have taken a small circular portion of it. I will show you a proper image also. Now just understand this. This is your DCT, this is your your full apparatus which has Bowman's capsule, afferent arterioles, glomerulus and efferent arterioles. Now students, what is happening? Okay, now what is happening? There are some muscle cells in your afferent arterioles, right? There are some muscle cells in your afferent arterioles. Say here, okay, some cells are there. These cells, what did I say? These smooth muscle cells, okay? These are smooth muscle cells. Everything has smooth muscle cells. All of you are aware of it. These smooth muscle cells will get modified into a type of cell called as JG cell. Will get modified into a type of cell called as JG cells. This JG cells is a type of an endocrine. What do endocrine glands do? What do endocrine as a whole system do? They will secrete hormones. Why is this modification happening? Because we realize that this person is undergoing through something. This person does not have salt. This person does not have water. Kuch to gadbad hai, right? Kuch to problem hai isme. So, what did they do? They took the afferent arteriole, some of the smooth muscles, modified itself to form JG cells, and that JG cells become endocrine. So, they will release a hormone. They will release a hormone. Okay. Now students, JG cells will release a hormone. Will it release all the time? No, it will not release all the time. When will it release? When the filtrate, what did I say? When the filtrate, okay, look here all of you, it is a little difficult concept, so I want all of you to have 125% concentration here. Students, so now what has happened? Afferent arterioles, some cells got modified, they became JG cells. They are ready to release a hormone. But should they release all the time? No, they should not release all the time. So, the filtrate is coming from here, right? The filtrate is coming, it is moving through your PCT, going to loop of Henle, it is going, it will reach DCT. Once it reaches DCT, DCT has a set of specialized cells called as macula densa. They will realize, is filtrate mein to kuch gadbad hai, right? It will understand that something is fishy in this filtrate. What is fishy in the filtrate? Maybe salt will be less, water will be more, water might be less, salt will be more. Something problem will be there in the filtrate. Now, every time you make chai, will it taste the same? Sometimes tea powder will be more, sometimes sugar will be more, sometimes water will be more, milk will be less, correct? Correct? Has anyone told every time, wow, what a chai? They'll be like, hey, it's not okay, it's okay today, right? Right? Students, yeah, I am talking Hindi because you people asked me. Now you people are telling don't talk Hindi. I am only telling gadbad word, so you remember. Gadbad word you remember because filtrate, okay, filtrate will have some problem. So students, what has happened? The filtrate from the glomerulus, it has come to PCT. It will move all the way, okay, it will move all the way to loop of Henle. It will finally reach DCT. Now, DCT has specialized cells called macula densa, okay, specialized cells called macula densa, which will understand that there is a problem in the filtrate, and it will give signal to whom? Okay, it will give signal to JG cells. Students, I only know gadbad word in Hindi. With that Hindi, I cannot survive. You know why I told gadbad is because when I was teaching in one class, I told this word gadbad. That student don't now also, whenever she sees me, she'll be like, ma'am, I still remember ma'am, macula densa because of the word gadbad. So, I thought it might work for you people. If it does not work, it's okay for me. Okay. Clear? So, students, this DCT will understand some problem is there. So, what it will do? It will tell JG cells, sorry, 1 second, yeah. Okay, it will tell JG cells, this macula densa, these cells are called macula densa. Okay, these cells are called Students, these cells are called macula densa. This macula densa will understand that filtrate has some problem. What could be the problem? Salt will be less, water will be less, salt will be more, water will be more, any of this problem. So, this macula densa will send message to whom? It will send message to JG cells telling that you release the hormone, and the hormone released is renin. The hormone released is renin, which will be able to solve all these problems. Clear to all of you? Clear to all of you? Yes, yes or no? So, let us go quickly once again. So, students, whenever there is a problem with our body or whenever there is a problem with our filtrate, imagine PCT, loop of Henle, someone did some problem. Okay, because of that the filtration did not go well, some salt was less, water was less. Now, for that we need a counter mechanism. We need to make sure that the person does not die, right? We need to make sure that the person does not faint. BP will be low, water will be low, problem will happen. Right, that time we have a apparatus called as Juxtaglomerular apparatus or your JGA, okay, JGA. Now, this is the apparatus which has Bowman's capsule, glomerulus, afferent arterioles, efferent arterioles. Now, this afferent arterioles, the smooth muscles of the afferent arterioles will get modified into a specialized type of cells called JG cells, called it JG cells. That JG cells will have an endocrine activity, that means it will be able to release a hormone, okay. Now, this JG cells will not release hormone just like that. It needs approval from whom? It needs approval from the DCT, because the filtrate will ultimately go to DCT. DCT and collecting duct will do conditional reabsorption. It will not do just like that reabsorption, right. So, when the filtrate goes to DCT, it will understand that some problem is there, okay, some problem is there. That means salt is less, water is more, some problem has happened. We need to fix the problem. So, this macula densa which is present inside the DCT special cells, they will send signal to JG cells telling you do one thing, you release renin. Rest of the job renin will do and renin will fix all this problem. Clear? Clear? Okay, clear as water. Thank you. So, students, can I tell macula densa is sensor cells? Yes, they are sensor cells because they are able to sense that some problem is happening. Clear? Very, very important concept. If it's not clear, please let me know. Okay, otherwise I can do it again once again for you people. Yes, okay. Did you all write it down? Did you all write it down? Because I just labeled and explained, I didn't write a lot of notes. Students, give me 1 minute, I'll just fill water and come. Okay, next topic is the most important topic. What is that? The main topic, right? The main plot twist of this chapter is your urine formation. So, students, look at this image. Okay, this is your image. Is it visible to all of you? Visible to all of you? Those who just came to the class, quickly like the session and mark your attendance. Students, this might look very complicated. I will just tell you which are the topics that you need to focus. Okay, students, here you have to focus DCT. Okay, distal convoluted tubule. You can see this is your macula densa. This portion is your macula densa. You can see here they have labeled it, and then you can see that afferent arterioles, some of the cells, okay, granular cells, you can see some of them have got modified and they will behave like your JG cells. Got it? They will behave like your JG cells. This is your glomerular capillaries. These circles that you see, this and all is not required for you. Just see this much. Just see this portion. Okay. Yes, all of you can just see. If it's confusing, let's not look into it. And here is your quiz. Ready for the quiz? Quiz, all of you who had 3 points. Now are you ready? Is it too bright? I don't know how to reduce the brightness. One second, students. I think this is fine. Is it visible? Too bright, no? Sorry, students, I put off white. Now I don't know how to edit this. Okay. I can't do anything also. Okay, never mind. Just adjust. Okay. Shall we start? One, two, three. 1, 2, 3. I hope it is visible. Done, done, done. Yes, one collecting duct, vasa recta, glomerulus. Okay. Yes, very good, very good, very good. Good job, good job. All of you can give yourself 3 marks. So, 1 is collecting duct. 1 is collecting duct. Okay. 2 is vasa recta. Okay, vasa recta. 3 is glomerulus. Okay, 3 is glomerulus. Good job. So, students, how many marks? All of you got 3. So, now we have completed 6 marks. Okay, 6 marks we have completed. Got it? 6 marks we have completed. Yes, very good. Okay. There's nothing to get confused. Vasa recta will be the one that is going to that, U-shaped structure. Here when it comes, it is peritubular capillaries. Clear? That's all. Nothing is confusing as if. Did you understand? Vasa recta is the one that will be extending to your loop of Henle. Okay. This here it will be your peritubular capillaries. Clear? 6 marks. Good job. So, when you comment after the session, do not forget to mention your marks. Even if you lose 1 mark, ma'am, 5 out of this much or 6 out of this much, no problem. I trust you people. So, I hope you will maintain the trust. Okay. This is loop of Henle. The yellow color tube is loop of Henle. This blue and red that you are seeing are the blood vessels. Clear? Okay. Yes. Yes. Now we are starting our main topic. Are we all having some energy? Can we see some fire emoji? Fire emoji. Yes, yes. Let me see if the energy is still there. All of you like the session also? Yes, very good. The energy is regained. Regained. Okay. So, students, urine formation has 3 steps. Okay. Urine formation has 3 steps. Okay. Step 1 is called your glomerular filtration. Glomerular filtration. Second one is called your tubular reabsorption. Tubular reabsorption. Third one is called your tubular secretion. Got it? Tubular. Yes, this lecture is more than enough for this entire chapter. Okay. So, we have urine formation happens in mainly 3 steps. Okay. Happens in 3 steps. Okay. Glomerular filtration. Okay. Tubular reabsorption, tubular secretion. These are the 3 steps. Now, let us understand where does all of this happen. Students, just looking at these names, can you tell me the locations? Just looking at these names, can you tell me the location? Where does first one happen? Where does first one happen? First one, glomerular filtration will happen where? Just with the name you can tell. Can you not tell with the name? Yes, glomerulus, very good. It will happen in the glomerulus. Okay. Tubular reabsorption and tubular secretion. From the name, what do you understand? What do you understand? Tubular. Tubular. Yes, it will happen in the renal tubules. It will happen in the renal tubules. Clear? Specific we will not go. We will not go to the specific. We will just write renal tubules is where your tubular reabsorption and tubular secretion will happen. Clear? Yes. One second. We are just drawing a structure to just show you, okay, how it is working. Okay. So, I have just drawn blood coming in and blood moving out. Okay. That is what I have drawn. Now, this is your glomerulus. All of you know that, right? So, students, look here. Let's look here. Now, we are going to understand where everything is happening. Now, blood is going to come in from the afferent arterioles. Agreed, all of you? Blood is going to come in, right? Blood is going to come in to your afferent arterioles. Here, glomerular filtration will happen and it will go out to your PCT. Correct? It will go out to your PCT. So, the filtrate, right, that is your glomerular filtrate will move to your tubes. Move to your tubes. Agreed, all of you? So, your first step will happen here. First glomerular filtration will happen here. After that, this is going to move into the tubes. I have just drawn the tube straight, so it is easy for our understanding. Easy for our understanding, okay. Now, here, what is going to happen? The filtrate is going to get reabsorbed, right? Filtrate is going to get reabsorbed. So, here, second step will happen. So, your glomerular filtrate, that is your GF, will move to the second step, and from the second step, whatever is less in the filtrate, will be given as secretions, right? That will be your step number 3. Clear to all of you? Clear to all of you? So, first step will happen in the glomerular filtration, second and third step will happen in your tubes. Clear? Clear? It is just the positioning I am showing you, nothing else. Nothing else I am teaching you, I am just telling you where and all, what and all is happening. Okay, here reabsorption is happening, that means from the filtrate we are reabsorbing things. Okay, here what is happening? We understood that filtrate has something less, so we are giving secretion is happening. Clear? Yes, even I need a break. I need a 5-10 minutes break. I cannot breathe. Okay, so shall we take a break? Students, a 10 minutes break. I need it. Okay, we will take a break. Those who don't want break, please write your notes. Yes, this is the topic. So, I will start from glomerular filtration in the next after the break. Okay. Now, what's the time? It's 8:16. 8:16. Yeah, we'll take a break. 8:17 it has happened. So, how long we'll take a break? Students, works? 8:30? 8:30 works, 13 minutes? Yes? Yeah, till 8:30, yes, as you people told. 9 is too late. We will just take a break when I need to sit for some time. Okay? Yes, quickly go, have some snacks, drink some tea or whatever, take some rest and come back. Okay? I will also take a quick rest. Dinner is not now, students. Dinner we'll have to have later. Okay, dinner break I will give you at I'll give off dinner break only directly. Then I'll give you dinner break. Wait, because since you people are going to eat faster, then I'll give you a dinner break. Then we will continue the session. Okay, 30 means 45. 8:45 works. All of you eat dinner and come. 8:45 works. Yes, yes. Okay, good, fine. Take a dinner break and come. Then we will not have break. Okay. Even I am back. Do you all have your lunch? I mean, sorry, dinner. Yes, let everyone join, then we'll start. Yes, many more are there. 100 students are missing students. So let's call them. No, I didn't have my dinner. I'll have later once the class get over. Yes, yes. Yes, are we back? Yes, most of you are back. Students, there are still some more people who have to come back. Okay, so yes, back again. Let me tell it in the telegram also. We'll tell them to be back. Students, did you all join my telegram channel? Gopika, Biology by Gopika. Quickly join Biology by Gopika. You can see my DP as well. It's basically, one second. Then this is the channel link. Okay. Students, tomorrow special class, do not forget to join. It is at 11. Okay, it is at 11 a.m. So do not forget to join. We will do a very good MCQ session. Yes, yes, some are coming back. Okay, so I think almost some will join slowly. So students, we were speaking about urine formation. So in urine formation, it takes place in 3 steps, right? Glomerular filtration, renal absorption and tubular reabsorption and tubular secretion, right? Now we are going to start the first part, okay? Peritubular capillaries and vasa recta, I will teach you at the end of the session. Okay, peritubular capillary, vasa recta is a type of a peritubular capillaries. I have explained this to all of you in my special class as well. Okay, peritubular capillaries are capillaries arising from the efferent arterioles. Okay, and vasa recta is a type of a peritubular capillaries. That's all. You don't have to know much about it. Just know it is a capillary rising from your efferent arterioles, and vasa recta is one among those. Okay, vasa recta is one among those. I remember explaining this in special class. They are basically tiny blood vessels. Okay, they are tiny blood vessels which is helping them to pass the blood easily. Clear? Okay. Yes, I told you I'll explain that meme at the end of the session. You have to wait till the end of the session. Do they tell the climax in the starting of the movie? Oh, this hero is dying, this heroine is going to die. Do they say that? No, right? They wait. You people wait till the climax. No. Okay. Yes, fine. Now, let's start with the first portion, that is your glomerular filtration. Students, I have drawn here a Bowman's capsule, okay, which is again inverted. Now, we have to understand the paths. We have to understand these paths to understand glomerular filtration, okay. So, understand in glomerular filtration, once the blood enters, okay, and once it enters the glomerular, it will be called a filtrate. Okay, filtrate. Okay, that means it will have all the substance which has to be removed, it will have water which has to be removed, reabsorbed, all this. Clear? Okay. Now, students, look here. We are going to understand this diagram. Okay, we are going to understand the diagram. The first outside layer, can any of you tell me what is this outside layer called? It is called the parietal layer. Okay, it is called the parietal layer. Did you people say it looks like a drilling machine? Okay. This is not exactly how the diagram is. I drew it like this. It looks little more calm. Okay. Okay. So, students, this is called the parietal layer. The outside layer is the parietal layer. The inner layer is called the visceral layer. Called the visceral layer. Okay. Now, your glomerulus happens in the endothelium. That means the glomerular filtration basically happens in 3 layers. Okay, it starts from the endothelium. It will go to the basement membrane. From basement membrane, it will go to the podocytes. From podocytes to the filtration slits. This you will see in your NCERT. Now, first we have to understand what is all this, right? What have I drawn all this? Okay, that is what we are trying to understand. Okay, so students here, okay, here if you see, this is your Bowman's capsule. This is your glomerulus. Okay, this is your glomerulus. One portion I have taken. Okay, glomerulus. Now, okay. Yeah, because I drew this, no, there cannot be copyright issue. This is my specialized art. I am the artist and only I can be the artist. Right? Thank you, students. Thank you for the motivation. Okay, now students understand. Okay, we are going to understand all this. There is a gap in between this, right? This is your endothelium. What is this? What is this? This snake water-like structure that I have drawn? Endothelium. Okay. This cells which I have drawn is your endothelium. Okay, endothelium. So, this blue color structure is your glomerulus. Inside your glomerulus, there is endothelium. Between the endothelium, there is going to be a space which is called the fenestra. How many of you know about the fenestra? The gap between the squamous endothelium, right? So, this which I have drawn here, they are endothelium, but they are not close to each other. Why? They are far from each other, because there is a space called the fenestra which is helping for glomerular filtration. Got it, all of you? Okay. All the spammers, all who joined late, quickly like the session. Do some help to the society. No, slit spores are completely different, Subhashni. You will study filtration slits when you come to podocytes. That I will show you. Fenestra is a gap between the squamous endothelium. Clear? It is a gap between the endothelium. Do not get it confused with filtration slits. Filtration slits is completely separate topic, separate part. Now you are studying only about the endothelium. So, understand your glomerular filtration happens from the endothelium. I will write it in one side. So, always you will understand what ma'am is talking about. So, it will happen with your endothelium. Endothelium specifically in your fenestra. Okay, endothelium to basement membrane. All of you look at your NCERT if all these 3 names are written. From basement membrane to your podocytes. In the podocyte, there are going to be filtration slits. Clear to all of you? No confusion? No confusion? Now, one second. I will show you that. Okay, students, look here, this is your NCERT. First we will see NCERT, then I think for you examples will be easy. So look here, urine formation has 3 processes mainly, okay. Let's first see your NCERT because most of you are not understanding this. Urine formation involves 3 main processes: glomerular filtration, reabsorption and secretion. The first step in urine formation is filtration of the blood, okay. Very important, that's what I told you. Once the blood enters, after once it enters the glomerulus, it's called a filtrate, right, which is carried out by the glomerulus and it is called as glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered out by kidney per minute, okay, which will constitute this and all I will explain you. Now we will only see this. The glomerular capillary blood pressure causes filtration of blood through 3 layers, very important, 3 layers. What are the layer? Endothelium. Understood why did I draw the endothelium? That water like structure, Christmas tree was standing, no, Christmas tree's reflection was coming on the water, it looks like that, no. You remember my drill like structure, it looks like a Christmas tree. Down what does it look like? It looks like water. That water blue color region which I have drawn is your glomerulus. Inside that what is there? Endothelium is there. Between the endothelium what is there? Fenestra is there, okay. So, the endothelium of the glomerular blood vessels, the epithelium of the Bowman's capsule, okay, this epithelium is where your podocytes will be present, okay, and your basement membrane, okay, basement membrane between these 2 layers, okay, between these 2 layers is where I drew the Christmas tree, understood, did you understand? Down is the basement membrane, top is the epithelium layer, between this whatever is there is going to help for glomerular filtration. Is this clear? If you do not understand this, you will not understand glomerular filtration at all, however the diagram is, okay. Clear students? Endothelium, epithelium of the Bowman's capsule, basement membrane, through these 3 layers, okay. In that between the basement membrane and the epithelium of the Bowman's capsule, whatever is present is your podocytes, filtration slits, your basement membrane, all these people are going to help for glomerular filtration. Got it? Got it? Yes? Endothelium, the water like structure, Christmas tree was standing, right? That Christmas tree is going to do what? That Christmas tree is the specialized cells, your podocytes. They are like this, they are interdigitating. That means, if I keep that drill like structure like this, they will get interdigitated. Got it all of you? Okay? That structure also is going to help. Clear? Now, since you understood this, look here. The epithelial cells of the Bowman's capsule called podocytes, okay, are arranged in an intricate manner. What is that intricate manner? This interdigitating manner, okay, intricate manner, so as to leave some minute spaces called the filtration slits or the slit pores. Clear to all of you? Clear to all of you? Yes, you will be able to understand Aruthar. Yeah, I can't read your name. Even if you are 11th standard student, you study this, you will understand. Yes, now I will explain the diagram once again. Now NCERT is clear. NCERT is clear. Okay. NCERT is clear to all of you. Okay. So, the glomerular capillary, that means the blood which is entering the glomerulus will go through 3 layers. Okay, what are the 3 layers? The first layer is your endothelium of the glomerular blood vessel. Okay, glomerular blood vessel, it is a blood vessel, that one's endothelium. Okay, then epithelium of the Bowman's capsule, which will have your podocytes. That podocytes are arranged in such a way that they will have minute gaps in between. Those are called filtration slits. Okay, which I will show you in the diagram. And then we have a basement membrane. So, the blood will go through these 3 membranes for filtration to happen. Done? No. Okay? Yes. Now, we will see the diagram. You will be able to understand. Okay, look here students. This is our Christmas tree. This is the water. Okay. Now, we are going to focus. What did our NCERT say? Our NCERT told that glomerular filtration is going to happen in 3 layers. First layer, endothelium of the glomerulus. This is the glomerulus. This is the endothelial cells. Can you all see? I am explaining. Calm down, Shanti. Okay, I am explaining. All of you just listen. Okay, it is, some concepts are difficult. I am trying to break it down as much as possible. Okay, students, what did the NCERT explain? 3 layers glomerular filtration will happen. What is the first layer? Endothelium. Endothelium, you can see the cells. These are endothelial cells. Squamous endothelium you have seen, right? These are endothelial cells. Between the endothelial cells, there are small gaps which are called as fenestra. They are also helping. Why I am mentioning fenestra is one of the years they had asked this. Okay, even though it is not a part of the NCERT, one time they asked this. That is the reason I am just mentioning it here. Okay, fenestra. Okay, now this lake you understood, endothelium is there, fenestra is there. This lake is whose lake? Glomerulus lake. Okay, who is the owner of the lake? Glomerulus. One layer of filtration clear. Okay, now we are going to the next layer. Okay, we finished lake, we saw lake and all. Now we are entering into the scenery, right? For the scenery to happen, okay, now the scenery to happen, what and all we need? We have to understand what are this Christmas tree like structure. This Christmas tree like structure are called as podocytes. What did NCERT say? The second layer is epithelium, right? Epithelium of the Bowman's capsule, right? Epithelium of the Bowman's capsule. So, now we are entering into the epithelium of Bowman's capsule which has podocytes. This Christmas tree is nothing but podocytes which are interdigitating. They are in an intricate manner, different manner, so that they will have space in between them. Clear to all of you? So, they have space in between them. So, that space will allow them to form your filtration slits. Can you see the space? Can you all see the space between them? Can you see the space? Filtration slits. Clear? Yes? This is glomerular layer, dear. This is glomerular. This is your Bowman's capsule. So, this is the epithelium of the Bowman's capsule. Okay, I will write it here. Epithelium, Bowman's capsule. So, from endothelium, we entered into the epithelium. In epithelium, who is there? Podocytes are there. Between podocytes, gaps are there, that is called as your filtration slits. Clear? Clear? Yes or no? I am going very slow. Very slow. Now, between this person, one person is there. Whom did we miss? Basement membrane we missed. Basement membrane. Where is basement membrane? Can anyone tell where is basement membrane? This is basement membrane. This gap which you see, no, between your endothelium and this one, that is your basement membrane. Clear? Is there any student who didn't understand? Thank you, Indu. Students, anyone who didn't understand? We have to go very slow. That's why I'm going really slow. Okay. I think I can draw one more diagram to show you one more thing very easily. Are you all with me? Okay. Students, what we were studying till now is this much of your glomerular filtration. Are you able to understand now? Are you able to understand? Yes? Okay. Endothelium, dear, not epithelium. Endothelium. Squamous endothelium, not epithelium. Epithelium of Bowman's capsule. Jyoti, clear? Okay. Students, this portion is what I have enlarged and drew here. Got it? This portion is what I enlarged and drew here, where I took a small portion of the Bowman's capsule, I took a small portion of the glomerulus. So, what did NCERT say? NCERT told that glomerular filtration happens in 3 layers. Okay, that means the blood which has come from the afferent arterioles, okay, from the afferent arterioles will first go through the filtration process by first going through the endothelial cells, okay, endothelium of the glomerulus. From there it will go to the basement membrane. From basement membrane it will go to the epithelium of Bowman's capsule which has podocytes. Okay. One topic only I will die of, students. Is this clear? Is this clear? Now clear? Okay. So what has happened? This small portion I have extended here, but in your NCERT, no diagram is given, only 3 words are given. With that 3 words, you will get 0. Okay, so that's why I have drew this and I have made you explain. Okay, so we are starting from glomerulus, which has endothelial cells, squamous endothelial cells. In that between gaps are there, which are called as fenestra. They are not filtration slits. Okay, from the endothelium, we are moving to this layer, which is between these two layers, basement membrane. From basement membrane, we are moving to podocytes, which are interdigitating. They are present in an intricate manner and they have gaps called as filtration slits. Okay, they have gaps called as filtration slits, which will help in filtration. Okay, so we cross the 3 layers. So this becomes the first layer, right? This becomes basement membrane, becomes the second layer, and this becomes the third layer. Clear? More than this, I don't think I can break it down. Okay, I don't think more than this I can break it down. Students, basement membrane, I have just drawn a diagram. There is no much, we do not know the cell arrangement as such. It is just to make you understand, because your NCERT has given only 3 words, but it is important for a teacher to break it down, right? If I just explain it like NCERT telling endothelium, epithelium, that, this, you will not understand. Got it? That is why I am explaining you with diagram. Okay? Yes. Okay, fine. See students, I will show you one more diagram for more understanding. Okay? More understanding. See, look here. Can all of you see this? Can you all see endothelial cells? See, this is your endothelium. Okay, this lake-like structure, no, is your endothelium. There, can you see this pink color endothelial cells? Between that, the gap, can you see fenestra? Fenestra. All of you focus here. If you give 5 minutes on this topic, you are done with the major chapter. 16 marks you will get from this chapter. So, it is not a game. We need to be serious. Okay. So, students, look here. This portion, glomerulus, we have endothelial cells. Between the gaps, we have fenestra. Then we have our basement membrane, which is written glomerular basement membrane. After the basement membrane, you can see these cells, right? These are your podocytes. Can you see how the podocytes are there? And the gaps between the podocytes are called as filtration slits. Can you see? Gaps are called filtration slits. Clear? Clear, clear? Okay. Then here you have urinary space, parietal epithelium, that and all is not required. So here epithelium comes, here endothelium comes. Clear? So do not get it confused. Here what you are talking about is your endothelium. Third layer which we are talking about is your epithelium of the Bowman's capsule. Clear? Okay, great. Fine. I have broken a big stone for you people. Okay. Wasn't it a big stone? It is a big thing to understand, no? Yes, but we have understood. Is there any student in my class who didn't understand? No, right? Did you all mark your attendance, all who are new to the class? Class. Yeah, you call the glomerulus, that area, as glycocalyx, which you do not have to know. Because again, I'll have to, like if we talk about glycocalyx, then we'll have to explain again about it. But NCERT does not tell us to explain so much. So, we are only standing to whatever is required. Okay. So, glycocalyx, let's not break our head. Now, students, glomerular filtration, what is it? First of all, it is a non-selective process. What do I mean by non-selective process? I do not care if in my class there are weak students, toppers, I do not care. I teach everyone equally. Even if one student asks me a doubt, I will explain it again. Get it? That means I'm not selective. I'm not like only toppers attend, or weak students don't. I'm not like that, right? Same way glomerulus also is non-selective process. That means it does not care if water is coming, sodium is coming, potassium is coming, carbonic acid is coming. They do not care. It only knows so much that if the membrane we studied, right? If the filtration is going to happen, if they are too large, they will be outside. If they are too small, they will enter. Got it? Got it? So, students, when you are filtering tea, right, when you are filtering tea, you will not care, oh, will this small ball of tea go inside, you will not care. But if you put a tea leaf, you know that tea leaf is going to be on the sieve and the liquid is going to come down, correct? So, same way this is a non-selective process, that means whatever is smaller particles will pass, larger particles will not pass, especially your proteins. Proteins are way too large, so they will not pass. Large, so they will not pass. Clear to all of you? Non-selective process, okay? Now students, here go, what is that? As if, what is that? No toppers, thank you, Dr. Strange. Yeah, Kirti, I am giving everything that is required for your NEET, dear. Like whichever questions, some words have come, I have explained that, like fenestra and all that, no? But whatever is there in your NCERT is the main focus. Clear? Okay, students, now we are going to study very important concepts. So, I want all of you to give me your 2 minutes of attention clearly here. Okay, beat yourself, get up from the chair, sit down, do something very, okay, very sharp it should be, your listening skills. Now students, I told you that it is a non-selective process. That means only the smaller and larger will do the variation. But one thing happens. If, okay, so before I explain that, I don't know how many times today I have drawn glomerulus, but even if you people call me glomerulus, I am fine with it. Okay, this is your glomerulus, correct? Students, all of you know that when something is passing, right, when something is passing, now you are standing for food in the canteen and you know that only first 100 people will get papad or first 100 people will get gulab jamun. You will stand in the gate in such a way that you are ready to push anyone and go over them, correct? Okay, when you all went for Leo movie, if the theatre was houseful and say first 100 people will pakka get the ticket, you would have pushed everyone, correct? Same way the blood also has to push, if blood pushes only, it is going to come out like this, no? It is going to enter after getting filtered, it has to move to the PCT. If the blood is like, oh no, tomorrow I will go, no problem, what tension. If it thinks like that, will it be able to move? No. So, there should be a pressure difference, right? Due to the pressure difference is the reason why the blood is able to move faster or slower, whatever, is because of the pressure difference, okay? What is happening? Okay, all of you stop fighting in the chat box, be kind. What is this? Okay, so students, due to the pressure difference, there can be an impact on the blood flow. Got it? So, that can be in a positive way. That means that is going to favor your glomerular filtration or favor the filtration. And this is going to be negative. That means they are not going to favor the filtration. They are going to give an opposite force. Newton's law, every action has an equal and opposite reaction. So, the action is this, means reaction will be this. Okay. So, students, first one, the positive force, that means which is allowing the blood to move into the glomerulus, is called as your glomerular hydrostatic pressure. What did I say? Glomerular hydrostatic pressure. Okay. This pressure is allowing, it is forcing the blood to enter the glomerulus and then go out as a filtrate to the PCT. Got it? So, glomerular hydrostatic pressure is giving positivity, or it is giving more pressure, telling that more blood go in. Okay, so more purification will happen. Got it, all of you? So, it is caused. How is this caused? How is this glomerular hydrostatic pressure caused? Okay, it is caused because of a difference in the diameter of your efferent and efferent arterioles. That means efferent arteriole will have more diameter, so more blood can come in. Okay, clear to all of you? Okay, clear? So, glomerular hydrostatic pressure is mainly because of the difference in the afferent and efferent arterioles. Okay, difference in the diameter of the efferent and afferent arterioles. Clear to all of you? Now, GHP can be somewhere, okay, GHP can be somewhere between, mostly it is given 60, okay, 60 mmHg, that is pressure. All of you understood, no? Glomerular hydrostatic pressure is the pressure due to which more blood will come from afferent, so we can go into more filtration. Clear? More the blood, more the, what is that, more the merrier, okay. So, more the better. Now, negative means they are giving force against this glomerular hydrostatic pressure, okay. It is telling how much of a pressure you give, I will give back the pressure, okay. So, it is giving negative, okay, negative. So, look here all of you, all my relatives, aunts, uncle, everyone look into this one, okay. Look here, first negative pressure is called BCOP, that means it is opposing the filtration, okay, it is opposing the filtration. And okay, it is opposing the filtration, and the first one BCOP, which is called your blood colloidal, okay, blood colloidal osmotic pressure, okay, blood colloidal osmotic pressure, okay. Clear to all of you? Clear to all of you? Blood colloidal osmotic pressure. Clear? That means it is giving pressure against this. It is giving pressure against this. Should I slow the speed? Yes, speed is okay, no? This is my normal speed. I have not even eaten dinner. So, I don't have that much speed also. So, students, what is happening? Blood colloidal osmotic pressure is the pressure exerted by colloids. All of you know that there is colloids in the blood. Yes or no? Yes or no? Is there colloids in the blood? Okay. So, it is what is this colloids going to do? It is going to block H2O from entering. What are the colloids going to do? Colloids are present in the blood. All of you know this. These colloids are going to block the water from entering inside. Now, if water does not enter, how will urine formation happen? Negative, negative. So, the most common colloid is your albumin. That is rich. Blood is rich in albumin, right? So, this albumin will stop the H2O from moving. So, that is a negative pressure. This is clear to all of you? Is it clear to all of you? It is blocking H2O from moving, especially colloids like albumin, which is causing a negative impact on the filtration. So, this BCOP is this, what it tell, is around 30 mmHg. 30 mmHg. Okay. Then, next one, two I told you, two negative forces are there. Second one is called your capsular hydrostatic pressure. Capsular hydrostatic pressure. Now, this is also a negative force. What is happening? Students, understand, there is already this Bowman's capsule, right? Bowman's capsule will already have some capsular filtration. Yes or no question? Okay? Yes or no question, students? Yeah? No, students, I'll have to go home and have my dinner. Now I'm in studio. Okay? Thank you so much for your concern. Students, now tell me, Bowman's capsule have some capsular filtration. Yes or no question? Yes or no? Quickly answer. Here, some filtration will be there. Filtrate will be there. Can I call it capsular filtrate because it is present in Bowman's capsule? Yes, very good. I'm asking, okay, I'm asking students, does your Bowman's capsule have some filtrate already. Before this glomerular filtration happening, they will have some kind of filtrate, no? Yes, right. Now that filtrate will be like, no need, no more blood inside. I don't want any more blood inside. I am already here. Now I am already a teacher here. If one more teacher come and teach the same chapter, will I be okay with it? I will not be okay with it, right? Same way, this capsular filtrate also will be like, I am already here. I will not allow any more filtrate to come in. So it will start putting pressure. It will start pushing the glomerular hydrostatic pressure. It will go against the glomerular hydrostatic pressure. Is this clear? Is this clear? Clear? Yes? Okay. So, students, capsular hydrostatic pressure value I will tell you, CHP is equal to 20 mm Hg. Okay, 20 mm Hg. Now, this is how much? Is there any student in my class who hasn't understood these two types of pressure? Please let me know. Very important concept and not very well explained in your NCERT. So, you need a teacher's help, otherwise you are not going to understand just by reading. If there is any student who is thinking I will read later and understand, you will not. Okay, you will not. All clear? All clear? Now, students, now we saw one positive, that is, we saw one positive pressure which is called your glomerular hydrostatic pressure, GHP, and we saw two negative pressures which is one BCOP and CHP, correct? Now, what are the values I gave you for each? All of you tell me. GCP, how much I give you? 60 mm Hg, right? BCOP, I gave you 30. We are going to do maths. Here I give you 20 mm Hg. Now, we have to calculate with all this opposing pressure and favoring pressure, how much pressure is actually glomerulus taking for moving the blood, correct? Correct? Let's do some maths. I will give you the formula, you will do the maths, okay? I don't want anyone to tell before that in the chat box. So, net, I can tell net glomerular, net filtration pressure, net filtration pressure, I am writing it here for students who will later watch recorded. Students, net filtration pressure is equal to GHP, okay, minus BCOP plus CHP. These two are negative, so we will put them in bracket, okay. Board mass rule and people remember. Now, tell me the answer. Tell me the answer. Not EPP, effective filtration pressure, EFP. You can tell EFP or NEP, NFP, sorry, NFP. Clear? Capsular filtration, Vaishu. Students, 1 second, one student is asking doubt. Students, capsular filtration means Bowman's capsule will already have some amount of filtrate. So, when new filtrate is coming or when new blood is coming, it will know that I have to go down. So, it will be like, no, I don't want to move anymore, don't need. So, it will put pressure back. So, the pressure exerted by your capillary filtration or filtrate, which is already there in Bowman's capsule, is called your capsular hydrostatic pressure. Clear, clear, Vaishnav? Vaishu, clear? Yes. No, this is in your NCERT. This is in your NCERT. Okay, yes. What is the calculation? So, GHP is 60. 60 minus 30 plus 20 gives 60 minus how much? 30 plus 20? 50? This is a MCQ question. Directly they will give you this number. So, that means the net glomerular pressure that is required for a blood to become a filtrate or for glomerular filtration to happen is 10 mm Hg. Clear? Okay, 10 mm Hg. NFP full form is net filtration pressure. That means how much pressure should be there for glomerular filtration to happen. So, we need 10 mmHg pressure for glomerular filtration to happen. Clear? Students, is nothing, it's like Newton's law. Every action has an equal and opposite reaction. The action is given by glomerular hydrostatic pressure, telling that let more blood go in, so more filtrate will be formed. Other two people will be like, no, I don't want to do any of this job, I will oppose you. So, this opposing and favoring will come into our valuation where we get 10 mmHg, which will favor your glomerular filtration. Okay? Yes. Okay. I hope it is clear. So, 10 mmHg is enough for glomerular filtration to happen without any tension. Okay? Yes. Now students, let's also understand few more things. Okay. Now, glomerular filtration, right? Glomerular filtration, what and all is there? Glomerular filtrate, sorry. Glomerular filtrate. What and all is present? Urea is there. Uric acid is there. Water is there. Amino acids are there. What else will be there? Extra, who is there? Okay. What else will be there, students? Glucose will be there. Ions will be there. If a person is having tablets, antibiotics will be there. All these things will be there, correct? All these things will be there, right? H2O will be there, glucose will be there, amino acids will be there, lot of ions will be there. In the ions only everything will come, sodium, potassium, everything will come, correct? Got it? Yes. So, students, now how much glomerular filtrate is formed? How much glomerular filtrate is formed? Around 180 liters are formed in a day. Okay, per day 180 liters are formed. Will 180 liters also form urine? It will not form urine. Only 1.5 to 1 to 1.5, 1 to 1.5 liters will form as urine. That means the rest is fully reabsorbed, right? The rest is fully reabsorbed back. Okay. So, here there is a small thing that I will tell you. Okay, small thing I have to tell you. So, how much is actually like the blood coming in, how much is it forming? Okay. So, students, when we talk about your renal blood flow, okay, that is mentioned in your NCRT, renal blood flow, around 1/5 of the cardiac flow. That much blood is, okay, cardiac flow. That means cardiac output is around say 5, okay. So, we will get around somewhere around 1100 to 1200. This is how much we have renal blood flow. That means this much blood is getting flowed into the kidney, correct? 1/5, okay. So, this much per minute, okay. Same way plasma also we have to calculate, okay, renal blood plasma. So, students, how much of our blood has plasma? I mean, how much plasma does our blood have? Okay. Renal plasma will be around 500, 600, right? Okay. Okay, how much will be? So, we will say approximate will be around 600 to 700. 600 to 700 per ml per minute, okay, per minute. From this only we calculated. Clear to all of you? Clear to all of you? Yes? So, glomerular filtrate around 125 ml is formed per minute. Out of that 180 liters are formed per day, out of which only 1 to 1.5 liters of urine we are throwing out. Clear? Throwing out. Okay? Yes. Okay. Let's go to the next topic, that is tubular reabsorption. Okay? So, we studied so much, no? Do you remember everything? Or did you all forget? Tubular reabsorption. So, students, when we talk about tubular reabsorption, I will draw the diagram on the side. So this is just to make you understand, okay? This is not the perfect diagram or anything. This is not even near to the actual diagram, just to make you understand, okay? So this is our normal nephron, okay? Okay. Why, bye ma'am and all. Study, where are you people going? Okay, so students understand, when you do tubular reabsorption, some of the things have to be reabsorbed. So how are they reabsorbed? They are reabsorbed depending on if they will be reabsorbed again or not. In the sense, can they be reabsorbed or no need? We do not need them that much, so we will reabsorb them little. Some things we don't need at all in our body, so we will just throw them. So depending on that, we have something called as high threshold, okay, low threshold, and we can say non-threshold, okay, non-threshold. Okay. Now, what do I mean by these? I mean that whichever has high threshold, that means whatever comes under high threshold will be maximum reabsorbed. That means maximum I can reabsorb them by the tubules, okay. Maximum reabsorption will happen. Low threshold means partial reabsorption. We cannot tell minimum, we will tell partial. Partial reabsorption. Here, no reabsorption at all, okay. No reabsorption at all. Clear? Okay. Okay, no reabsorption at all. So, maximum reabsorption, who and all will be reabsorbed? Glucose, water, NaCl, amino acids. So, we will write here. Students, again this could be a question in your NEET. Okay, glucose, Na, H2O, amino acids, right? These things will be reabsorbed. Maximum reabsorption have to happen because these are our most important things. We cannot lose them. Correct? Got it all of you? Okay, got it all of you? Okay, here partial reabsorption means urea. We take partially urea for what? Why do we take partially urea? Students, high threshold means something which we can maximum reabsorption can happen. Low threshold means partial reabsorption will happen. Non-threshold means complete not at all. We will not at all reabsorb them because they are useless for our body. Okay. So, in maximum reabsorption, we are taking in glucose, sodium, water, amino acids which are very, very essential for our body. In partial, we are taking urea and uric acid. Urea and uric acid. Urea we are taking for osmoregulation, very good, most of you told the answer. And uric acid we are getting some of them because of metabolism of purines. Okay. Non-threshold means your creatinine which is not at all reabsorbed. Okay. Not reabsorbed at all. At all. Okay. Creatinine. Okay. Creatinine. Clear? Clear to all of you? Nothing very complicated. Just understand some things have to be maximum reabsorbed, some we do not have to. Some we do not have to. Clear? Clear? Okay. Now students, see, look here. Your osmolarity of the blood is around 30. 300. Okay, 300. So that blood will enter, okay, around 300. It will go through your PCT. Okay, it will go through your PCT. It will still be 300. That means, can I say in the PCT it is isotonic? Can I say in the PCT it is isotonic? Yes? Yes? No? So, students, it is isotonic here. That means no much change. Okay, so let's see what and all is getting reabsorbed. I told you NaCl is getting reabsorbed, HCO3 is getting reabsorbed, water is getting reabsorbed. Okay, all this is getting reabsorbed, correct? All of you check your diagram in your NCRT. Okay, K plus is getting reabsorbed, amino acids are getting reabsorbed. I don't have place to write everything, but amino acids are getting reabsorbed. So, students, in tubular reabsorption, you only have to understand about, you only have to understand about your what and all is getting reabsorbed. You don't have to know a lot. Okay. One second, students. Okay. So, only reabsorption, what and all is getting reabsorbed, you have to know. Okay. So, what did I say? 300 osmolarity blood that is coming in will remain, osmolarity 300 only will remain. So, here in the PCT, no differentiation of the mole has improved. That means no salt is added or anything, everything is going reabsorbed. Now, when we come to loop of Henle, water is getting reabsorbed, correct? Maximum reabsorption of water is happening. Students, when water goes out, will concentration increase or decrease? Will it become hypertonic or isotonic? All of you tell me. Tell me, if I continuously remove water, will the osmolarity increase or decrease? Will it become hypertonic or isotonic? Very good, Kaustuba. I cannot read your name. Nikita, Subhashni, Nandini, Sandvi, yes, Preeti, Ruth. Okay, very good. Hypertonic, it will become hypertonic. Here it was isotonic, right? Because I wasn't removing any water, I was actually removing all the ions. So, it was actually becoming normal. Here it will become hypertonic, correct? Here it will become hypertonic in this loop of Henle. So, students, we are actually studying what and all is getting reabsorbed in which and all parts of the tube. Okay. So, in PCT, Na, HCO3, amino acid, H2O, potassium, all of this is getting reabsorbed. When we come to your descending loop of Henle, water is getting reabsorbed. Okay. Clear to all of you? Now, when we talk this part, okay. So, here my solution became hypertonic. Okay. Here it was isotonic. Okay, here it became isotonic. Here what is happening? Some things are coming out here also, that is nothing but your Na, K plus, Cl minus. All these people are coming out here, and here whenever these things are coming out, this will be passive reabsorption. That means no energy will be used. Okay, no energy will be used. Here calcium is going to come out. Okay, here it will be active. To remove out calcium, we need some energy. Okay, energy. Now students, it will slowly enter DCT. Will DCT and collecting duct do any job now? All of you tell me. Will DCT and collecting duct do any job? These two people, will they do any job? They will not do any job. Why they will not do any job? They will not do any job because they are going to do only conditional reabsorption. They will do conditional reabsorption. That means only during stress condition or when too much necessity is there, they will reabsorb. Clear to all of you? Clear to all of you? Right? Conditional reabsorption will happen. Okay? And collecting duct will give out some urea. Correct? Collecting duct will give urea. So, I can call it urine only when it reaches the collecting duct. Here everything I am calling it filtrate. Clear? Clear to all of you? Students, you have to know the diagram. You don't have so many things to know, just diagram. Okay, just diagram. Now, one very important thing I'll just inform you here. Students, so here PCT mein, in PCT what and all happened? Na, HCO3, amino acid, H2O, potassium, whatever you can remember, please write it here because most of the ions and things are getting removed here. Okay. Now, osmolarity of blood was 300, it will remain 300 here, isotonic. As it comes here you have to know something. Descending limb of loop of Henle, descending loop of Henle is permeable to water, and impermeable, it is impermeable to ions or it is impermeable to your salts. Okay. Clear? That is why it is only removing water here. Got it all of you? Now, ascending loop of Henle is impermeable to water but permeable to salt. Did you all understand that? Did you all understand that? Easy, no? Easy, no? Yes. Okay. These two people will do conditional reabsorption. Who and all? DCT and collecting duct will do conditional reabsorption when? Now, ADH is doing some job or some water is lost during stress condition, they will do this. Clear? Okay. So, quick recap. Here blood has entered, some of the things are getting reabsorbed. in the PCT, and that will remain, the blood will or the filtrate will remain isotonic. Okay, as it comes down to your loop of Henle, your descending loop of Henle is permeable to water and it will only remove out water. Okay, remove out, reabsorption of water will take place. As it comes, so because of that the solution will become hypertonic. As it moves forward, when it reaches ascending loop of Henle, it is impermeable to water and permeable to salts. So, it will only remove the salts or the ions, and it will move forward, and DCT and collecting duct will not do any job until the condition is a stress condition, because they are only there for conditional reabsorption, and I can call it urine only when it reaches the collecting duct. Clear? Okay, that is all about this. Okay, I finished it here only, students. See, this is the diagram. You can look here. See, whatever I wrote is here. Okay, H2O, H2O, here you can see NaCl, NaCl, distal convoluted tubule. In case they ask, these are the things that come out. Okay, active transport, students, see, active transport, this portion will do active transport. Okay, here also active transport. Here few things, NaCl active transport, H plus active transport, HCO3 active transport. That means they require energy for doing anything. This I explained. 1 second, students, I will just see. Here it is the reabsorption. Okay. So, reabsorbed by the renal tubule, this process is called reabsorption. The tubular epithelial cells in the different segments of the nephron perform this either by active or passive mechanism. Active and passive means ATP is used in active, passive means no ATP. Okay. And reabsorption of water also occurs passively in the initial segments of the nephron. During urine formation, the tubular cells secrete substances like H plus and K plus and ammonia into the filtrate. Tubular secretion is also important step of urine formation. Students, here they are not explaining much, they are just telling here and there. Okay. So, I will explain it to you. Okay, I will explain it to you. Oh no, students, 1 second, I didn't add one slide. Students, this is actually tubular secretion. Okay, this I will cancel. Okay, this is secretion. Error happened. Okay. Can we go to tubular secretion? Yes or no? Students, we will finish it in maybe an hour. Okay, another 1 hour maybe it will take. We have some more mechanisms to do. Okay. Yes. Okay. Now, tubular secretion means what? If the pH is disturbed, right? If the pH is disturbed, what is going to happen? We need to secrete some things out, right? So, according to your textbook, very less is informed. So, I will just write few things. PCT will secrete. Okay, what will PCT do? Okay, PCT will secrete H plus and NH4 ions. Okay, NH4 ions. Why they are doing this? To maintain the pH. Okay, to maintain the pH. Okay. Now, PCT is going to put H plus and NH4 ions. Okay, they are going to secrete. Now, DCT is going to do what? DCT also is going to give H plus, NH4 plus and K plus ion, potassium ions. Okay, you don't have much to know about this, you have just little bit to know about this. Okay, so I am giving you that little. Okay, collecting duct also will secrete, H plus, because students, we have to maintain the acid and the base pH. Remember chemistry, you remember, acid and bases, ammonium ions. Okay. So, this is mainly done to maintain the pH, and also when we tell maintain the pH, that means what? Acid and base balance. This is all happening where? It is happening in PCT, DCT and CD, that means collecting duct. It will not happen in loop of Henle. It will not happen. Secretion is happening here and all, PCT, DCT, CD, only when there is a difference in the pH. When they see that there is some problem in the pH, then only they will secrete. Clear? Isotonic or it will go to the DCT and collecting duct as isotonic. Only in the loop of Henle it will become hypertonic. When it reaches your ascending lymph, it will again become isotonic. In the DCT, it will become isotonic, because if it becomes hypertonic, then we are going to suffer, right? Okay, Praveen, I hope that is clear. Students, that's all. Okay, this is all about it. Your tubular secretion explanation is very less. Okay, but you should just know what is being secreted by each person. Okay. Now, next very important topic is your countercurrent mechanism. Okay. So, when I talk about countercurrent mechanism, I am basically talking about two tubes. Are you all sleepy? Can we have some fire emoji in the chat box? So, I also get the energy, because my energy is coming down. Okay. So, let's build each other's energy. Yes. Okay. Yes. Students, it's not tough. Let's see what is happening here. So, countercurrent mechanism means, when I talk about countercurrent mechanism, I am basically talking about conditions when there is stress conditions. So, in stress condition, which is the nephron that is going to help? The nephron that is going to help is your juxtamedullary nephron, right? Students, I am more tired than you. I haven't even had dinner. Come on, you people cannot be tired. Yeah, I am standing here continuously. So, you people shouldn't be tired. You people should give more energy to ma'am. Correct? Should give more energy to ma'am. More love to ma'am. I didn't have dinner. You people had dinner. You people had coffee, tea. I didn't have any of this. Only water. Only water. So, I want all of you to keep the energy. I don't want any of you to tell ma'am I'm tired. No, 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 no. Okay. Very good. Juxtamedullary nephron, right? So, when the condition becomes bad, our juxtamedullary will do some job, that is to help us with the stress conditions, right? So, during this, what is going to happen? We are going to take NaCl and urea into the medullary interstitium. Students, what is happening basically? In stress condition, the body will not have water. Correct? So, we are trying to reabsorb as much water as possible. If we need to reabsorb as much water as possible, that means we have to make the this medullary interstitium hypertonic. All of you know osmosis. Water has to move from a region of higher concentration to lower concentration. That means here we have to increase, right? We have to increase and make it hypertonic. So, to make sure that the water is not lost, right, water is not lost, we have to maintain it. For that, what are we going to do? We are going to make sure this medullary interstitium, right, I will show you which is medullary interstitium. Students, this is your medullary interstitium, okay, the gap between is your medullary interstitium, right. For that, we have to make sure that the sodium and urea is more there. Now, how does this happen? How does this happen? Basically, we are taking back the water, we are reabsorbing the water so that water does not get lost. Okay. For that, we have to do this mechanism. Now, what is going to happen? Now, this is AVR. AVR stands for your ascending vasa recta. What does this stands for? Ascending vasa recta. All of you make a note. This is descending vasa recta, descending loop of Henle, ascending loop of Henle. Now, what is countercurrent mechanism? Countercurrent mechanism means when two things are in high proximity with each other. Two tubes are in high proximity with each other, they will run opposite. They will run opposite. Okay. So, here you can see filtrate is going downwards, okay, going upwards. Vasa recta, blood is going, okay. So, here can you see the arrow marks have to be opposite. Here the blood is going up, here the blood is going down. Okay. Now, let us break it down, very simple, do not get it confused, very simple. Students, all of you like the session, all the spammers also like the session, okay. Students, what is going to happen? We know that in the ascending loop of Henle, NaCl is going to come out. NaCl is going to come out. All of you know, just now I taught you NaCl will be coming out. That NaCl will be picked up by your vasa recta and it will carry it in the form with the blood. It will carry it with the blood and it will carry all the way, the NaCl it will carry all the way and it will dump it here. It will dump the NaCl in the medullary interstitial fluid. Okay, I am writing here medullary interstitium. Okay, medullary interstitium. Okay, yes. So, now understood, the NaCl that came off ascending loop of Henle will be taken by the descending vasa recta and bring it till here and drop it down in the between. Okay, drop it down in the between. Now, I told you one more person we have to bring. Whom will we bring? Urea. Now, urea from where are we going to bring? We are going to bring it from collecting duct because we know that urea will be only given by collecting duct. Okay, this is collecting duct. It will give out urea. That urea also we will take it inside. Okay, that urea also we will take it inside here. Okay, so here also urea formation will happen. Now, you people tell me, now did we concentrate the medullary interstitium? We concentrated the medullary interstitium. Now, what are we expecting? We are expecting maximum water to come out. We are expecting maximum water to come out. Clear to all of you? Yes or no? Yes or no? Okay, students, look here, there is no confusion. In very stress condition, we need to make sure that maximum water gets reabsorbed. For that, we have to make sure that this medullary interstitium, now what am I talking about? Look here. One second. Yeah, what is this? This is your medullary interstitium. Now see students, if I draw blood vessels over this, this is your medullary interstitium. Between this, where reabsorption has to happen, correct? Okay, reabsorption has to happen. So, that medullary interstitium I have to make sure that it becomes hypertonic. So, my water, one second students, yeah. So, water gets maximum reabsorbed. Now, for that to happen, I need NaCl and I need urea. NaCl I am going to get from ascending loop of Henle. Okay, ascending loop of Henle will give out NaCl. That NaCl will be taken up by your descending vasa recta and bring it all the way till your ascending vasa recta and deposit in the medullary interstitium. Clear to all of you? Clear to all of you? Now, where am I going to get urea from? I am going to get urea from the collecting duct. The urea that is coming out of the collecting duct will be taken back into the medullary interstitium. Make this concentration, make it complex, okay, and reabsorb as much as water. Yes, clear? Reabsorb as much as water. One second, students. One second. Okay. Easy, easy, nothing, just so much only. I have to make sure this is the medullary interstitium has to get concentrated with salts. So, for that I am going to take NaCl and urea's help. NaCl will come out of the ascending loop of Henle. That will be taken by descending vasa recta. Taken all the way, dropped it into the medullary interstitium. Then, where am I going to get urea? I am going to take urea from the collecting duct. Like that, I made sure that this area is hypertonic, full of salts. So, what will happen? Water will move, water will come out, I will get more water. Correct? Got it? Maximum reabsorption has happened. This will happen during the stress condition. Clear? Okay? Clear to all of you? So, now we have to know how many countercurrents are happening, right? How many countercurrents are happening? One countercurrent is between this person, these two people, right? Ascending vasa recta, descending loop of Henle. Oh. Yes. No, that urea will not. Students, remember I told you about the urea that was used for osmoregulation. Don't you think this is osmolarity? To maintain the osmolarity, I am taking back some urea. Remember, are you able to connect everything? Are you able to connect everything? Huh? Yes or no? That is why I taught you in the starting itself. I taught you in the starting itself why we are going to take some urea. Okay, I took some urea back because I needed it for osmolarity, to maintain the osmolarity. AVR ascending vasa recta, DVR descending vasa recta. Okay, descending vasa recta. Here it is LOH, ALOH is ascending loop of Henle. Right, ascending loop of Henle. Clear to all of you? Clear? So, it is this clear countercurrent mechanism. Now, we have to write how many countercurrent mechanism is happening. Can any of you think and tell me how many will be happening? Countercurrent mechanism means two tubes which are in close proximity, one is going ulta, one is going downwards. Like one is going up, one is going down. Like you and your sibling, one will be very silent, one will be very violent. One will be very punctual, one will be not at all punctual. Right? So, how many countercurrent mechanisms? Let us make a note. Okay? First countercurrent mechanism, it is between AVR and DLOH, because blood is going up, filtrate is moving down. First countercurrent mechanism, agreed all of you? Yes. Second countercurrent mechanism, ascending loop of Henle, filtrate is going up, blood is going down. This two. Second, agreed? Third one is between AVR, DVR, DLOH, ALOH. Okay, so I am going to write here your three countercurrent mechanisms. Okay, three countercurrent mechanisms are AVR and DLOH. Okay, first countercurrent, then DVR and ALOH, and between DVR, AVR, ALOH, DLOH. So, this becomes third one, okay. This becomes second one and this becomes the first one, okay. Got it all of you? 1, 2, 3. 1, 2, 3. Okay. Gamer Ammu is in BC, before Christ. Now she is asking when did the class start. We are coming to an end of the class. Students, clear countercurrent mechanism? Yes. Okay. My leg needs a break. I don't need, but my leg needs. By then write it down quickly. No one is going to write and give you all this. It is not given in your NCERT. Only 2-3 lines are given. All of you write it please. Yes, okay, fine. All the screenshot babas are here. They will take screenshot and later they will only not know where and all they have their screenshots. Okay. I hope it is clear. Countercurrent mechanism. Okay. Students, look here, one more thing I have to tell. Now, I told you a person who is eating a lot of meat, a person who eats a lot of meat, non-veg, okay, lot of non-veg, they will have lot of urea in their body. Agreed all of you? Yes or no? If more urea is taken in, then don't you think reabsorption also of water also will be more? Yes? Yes? Students, what I am trying to say, a person eats a lot of meat, okay, proteins. If a person eats a lot of proteins, that means urea content also will be high, okay. Urea content will be high because proteins are made up of amino acids. When urea becomes high, what happens in our countercurrent mechanism? All the urea will move to the interstitial medullary interstitium. More water will be absorbed. This person will get dehydrated. So, when you are eating a lot of meat, you need to drink more water. Okay. Health tip. With class, I am also giving you health tip. See what and all I am giving you people. Okay, students, hormonal regulation. For this, you people should really have peace of mind. Okay. Yes. Thank you, thank you. Sanvi, I don't know Sanvi, they might have felt like no need to give again headache to students, so they considered. Because actually, if you look at it as a mechanism, they are not in close proximity, but they are flowing up and down, correct? So, when you are looking at countercurrent mechanism, it is basically close proximity where one filtrate is going down, blood is going up. So, when we see vasa recta and loop of Henle or whatever, they each other are doing that, so it is helpful. But to be technically right, they are not actually close proximity. Got it? Right? Yes. Okay. Can we start? My leg is gone, students. I don't know what is happening, but leg is not cooperating very well. Okay. Students ready? RAS mechanism. We have energy. Need some exercise. Need some exercise. Yes or no? Ready? Okay, fine. Let's start RAS mechanism. Okay, that is called hormonal regulation of the kidney. Okay, hormonal regulation of the kidney means what? That means some hormones are released to regulate and make sure that the filtrate, everything is proper. The first mechanism is called as RAS mechanism. Okay, it sounds like a beautiful girl's name, no? RAS mechanism. No, fecal matter is not. Pavan, you have to see the starting of the class. Go back. Go back to the starting of the class. Very late question. Thank you friends forever. Thank you so much. Students, RAS mechanism full form. Renin angiotensin aldosterone. Okay. Renin angiotensin aldosterone system. This is the full form. Now, what is this person going to do? What is this person going to do? Okay. So, students, we notice that in juxtamedullary, okay, we are going to revise. Okay, you people will answer, I will not tell anything. Tell me JGA, what is the specialized cell formed from afferent arterioles? Question round. Question round. Okay. In juxtaglomerular apparatus, that is your JGA apparatus, what is the specialized cells called? Specialized cells. Good. JG cells. Correct? JG cells. Now, who is going to give signal to JG cells? Who is going to signal JG cells that there is some problem in the filtrate? Did you all like the session? Let me see. Attendance. Time for attendance. Let me check your attendance. All of you liked the session? Anyone bunking the class? Students, no bunking. Bunking is not allowed. Let's see. Yeah, not enough, not enough. All of you haven't liked. Quickly like. Attendance check is over. Yes, very good. Macula densa will give the signal. Okay. Macula densa will give the signal. Macula densa will give the signal to JG cells telling that you release whom? Renin. Now we are going to understand. Now we will understand what this boy was doing all this while. I told you, correct? Oh, why we should like, ma'am? Someone please give him reply. Why we should like the session? Someone please give reply. Okay. So students, listen. JG cells release renin. Okay. Okay. So my students have the best reply in the world. Look at them replying. Wow, wow. I'm so proud. I didn't have to reply. My sarcasm didn't have an answer, but they have. Okay, so students, okay, fine. So JG cells release renin. Now we are going to actually understand what is this. Students, so hormonal regulation in kidney means I told you that because the filtrate or some problem is there in the body, that is when these mechanisms will work. Now let's see what is the problem that could come. I already discussed with you that time that when a person is having BP low, okay, imagine the BP is low for the person, okay, or the blood volume, okay, blood volume is low, okay, imagine blood the volume is low. If blood volume is low, then that means blood water level also is low, okay, all these reasons. Yes, correct. GFR is low, GFR is low, that means glomerular filtrate rate also is low. These are the problems that a person can undergo. Normal, no, BP low, blood low, water low, all this is normal, correct. Your BP gets low when I told you in the assembly, correct. Now students, when these things happen, there should be someone to control or help with us. That is what renin is going to do. Now renin cannot directly go and do all these problems. So renin will go through many procedures. Now all of you focus here, all of you focus here. Okay, all of you focus here. Listen, renin is the hormone released. Now, renin cannot fix half of the problems. What did I say? Renin cannot fix half of the problems. Okay, one second. Yeah. Because renin cannot fix the problem, renin will not just be like that. Renin will be sent into the blood plasma. Okay, imagine this is the blood. Okay, it will be sent into the plasma. Okay, blood plasma. Renin will be taken into the blood plasma. In the plasma, there is going to be one person. Who is that person? Okay, we are going to see. Okay, the person is none other than angiotensinogen. Okay, angiotensinogen, which is released from the liver. Okay, so this is our liver. Okay, liver will secrete something called as angiotensinogen. Now, angiotensinogen is in the inactive form. That means it is not good for anything at this point. It is not going to do any job. So, who do you think will help? Who do you think will help? Renin will help. Renin will make sure this angiotensinogen gets activated. Are you all understanding? Renin cannot fix all this problem. But renin got an idea that angiotensin can fix all this problem. So, what will I do? I will activate angiotensinogen, which is released by the liver into the plasma. Clear? Yes? But angiotensinogen is a very strong person. It will not get activated just by renin. So, it will be like, okay, fine. Since you tried, I will help you. And it will become angiotensin 1. Okay? It will become angiotensin 1. Still it is not activated. It is very strong, no, full government is there, no, that attitude is there, no. just by small renin, I will not get activated. So, it will become angiotensin 1 and still become inactive. Now, will renin leave? Renin has to fix all these problems. Renin has so many tensions in life. Renin don't have time for drama. So, renin will do what? Renin, after angiotensinogen is 1, which is in the half inactive stage will happen, we are going to take the help of lungs. Whose help? We are going to bring new people into the system. Lungs we will take. Lungs will come, okay. Students, first time you will see orange color lungs. Okay, this is a golden opportunity that I am giving you. Okay, lungs is going to release something called as ACE. Can any of you tell me what is ACE full form? ACE full form? Okay, ACET. Okay, angiotensin converting enzyme, very good Subhashni. So, this ACE is none other than angiotensin converting enzyme. Now, angiotensinogen has to lose its attitude. Why? Because lungs also came. Now, it cannot be like, oh no, I will not. It will finally get into an activated form, that is angiotensin 2. Okay, angiotensin 2. Got it all of you? 1 second students. Yes, angiotensin 2. Now, this angiotensin 2 is capable of doing what and all? It is capable of fixing all these problems. How it is going to fix all these problems? Okay. Now, I realize that I don't have place. Okay, students, we will erase this and we will fix the problems. Okay. Angiotensin 2 will do many jobs. Okay, what are the jobs that it will do? Okay, angiotensin 2 will, okay, look here, all of you look here. Angiotensin 2 will fix all the problem. First problem it will fix H2O reabsorption. H2O reabsorption. That will, first job it will do is to reabsorb all the water. Now, when water is reabsorbed, automatically our 2 problems also will go. What are the 2 problems? Blood volume will increase. Blood volume will increase. Water also will increase. Correct? 2 problems sorted. Correct? 2 problems fixed. Now, next problem angiotensin will fix is we don't have GFR, right? We have less GFR. Okay. Now, that will be fixed. More pressure means what? Vasocontraction of your efferent arterioles. So, it will do vasoconstriction. Vasoconstriction means reduce the size so that more GFR will be formed. Okay, right? Vasoconstriction. Okay, so because of vasoconstriction, what will happen? More pressure will be felt. So, more pressure means more GFR. Did we fix one more problem? We fixed again one more problem. Okay, we fixed one more problem. Now, we have one more problem. What is the problem? BP. BP, right? BP we didn't fix. BP is still low. That person is still dying. Okay, so we have to fix that person. Stimulate aldosterone. That's why the name is aldosterone system. So, it will, again my problems are not solving at all, of no space. I think if they give me one whole wall of board also, I will still not have space. I don't know why. Students, you have to adjust with all my writings. Wherever I write, you will have to understand. Even I don't know why, yes. Okay, students, look here. So, vasoconstriction, okay, here we fixed our pressure. Next is BP. Okay, so, angiotensin will stimulate adrenal cortex. Oh, I don't think this is a good idea. Students, 1 second. Okay. Right here only. Students, what did I tell? So, the last thing that your angiotensin 2 will do, is to make sure it will stimulate adrenal cortex, okay. It will stimulate adrenal cortex to release aldosterone, okay, to release aldosterone, which will automatically help in conditional reabsorption of sodium, which will help in conditional reabsorption of Na. If Na increases, that means BP will increase. So, our BP problem also increased. So, did we fix all these issues? Yes or no? Students, please don't spam, yaar. Please don't spam. I'll be forced to teach you biology and I don't want to teach you biology. All the spammers. Yes, okay. So, look here all of you. Let's quickly see what RAS mechanism is all about. So, students, RAS mechanism or your hormonal regulation takes place when there is a problem. Now, what is the problem? Problem could be when the BP is low or the blood volume is low, H2O is low. When these things come, we already understood that our juxtaglomerular apparatus will release JG cells, right? Specialized cells called JG cells. That JG cells will release renin. That renin will not be able to help in solving all these problems. So, renin will instead go into the blood plasma and liver will secrete angiotensinogen, which is in the inactivated form, okay, which is inactive. So, renin will try to activate this. Now, this angiotensinogen will not get completely activated, it will get partially activated to angiotensin 1. Now, angiotensin 1, again we are going to take an enzyme from lungs which is called the ACE enzyme, ACE, angiotensin converting enzyme will get converted to angiotensin 2. Now, this angiotensin 2 will be able to solve all the problems. What are the problems it will solve? It will solve the problem by first reabsorbing H2O. When it reabsorbs H2O, blood volume and blood water level will increase automatically. Vasoconstriction of the efferent arterioles, that means increasing the efferent arterioles, right? When they reduce efferent arterioles, that means the blood that will go out will be less. So, efferent arterioles will start taking in more blood, okay? Vasoconstriction, which will automatically increase more GFR rate, okay? Then the last thing that they can do is conditional reabsorption. That means angiotensin 2 will stimulate adrenal cortex to release aldosterone. That aldosterone will make sure that conditional reabsorption, that means with the help of DCT, it will make sure that sodium is reabsorbed. Okay, they will only work during stress conditions. So, aldosterone will make sure DCT reabsorbs sodium and BP will automatically increase. Okay, clear? Samir, is it clear? Samir, clear? Conditional reabsorption? Yes, okay. Okay. I hope it's clear. Yeah, 5 minutes else it, students. Yes, Soumya, yes, yes. . Students, you can join my Telegram channel where you are allowed to text. You can text me on that. Okay. Yes. Okay. So, we'll finish off hormonal regulation. Okay. Because then it will get too late also. Okay. Yes. Thank you, Laharika. Okay. Students, next two more hormonal regulation, hormonal regulation of kidney is ADH. Okay. ADH means your anti-diuretic hormone. Okay. What is it called? Anti-diuretic hormone. Okay. Anti-diuretic hormone. Students, what is going to happen here? Anti-diuretic means to prevent the loss of water. Diuresis, very, very important. Diuresis means loss of water. Diuresis means loss of water. To avoid any loss of water, we have this anti-diuretic hormone. Clear to all of you? Clear to all of you? A person who is suffering from diabetics, they will have a lot of loss of water. In that condition, what is the mechanism that we need? We need ADH mechanism. Clear? Okay. Now, this anti-diuretic hormone is synthesized by hypothalamus. Synthesized by hypothalamus. Okay. And it is released by the posterior pituitary. Why you are not able to join, Soumya? Soumya, you can text one of them, they will send you the link. Okay, or in my community page it is there. Special class, no, community page I have put the link. Okay, students, it is released by posterior pituitary. It is released by the posterior pituitary gland. Got it? Pituitary gland. Okay. Now, what is actually happening? How is ADH going to help? Okay, this ADH will make sure, okay, now what is happening? Loss of water is happening. ADH will first try to activate your thirst. Now, I am feeling to drink water. Why? Because the ADH not in my condition, in this kind of condition, what is going to happen? It is going to activate your thirst center. It is going to activate your thirst center. Now, thirst center, what will it do? That means it will tell you to drink water. It will tell you to exogenously intake of water. Got it, all of you? We are trying to avoid loss of water. So, the first thing ADH will do is going to activate your thirst center by telling you that you are thirsty, drink some water. Okay, that is an exogenous process. That means it's happening outside, right? We are taking the help of a person. Now, second thing that ADH will do, first thing understood? Okay, second thing it will do is stimulate conditional reabsorption. This is what I told you people sometime back, conditional reabsorption. It will stimulate conditional reabsorption. Okay, clear? With the help of who? DCT and LOH, loop of Henle. So, now what will happen? The urine will get concentrated, so the person will not suffer from loss of water. Clear? Easy, no? Nothing is there much. One will activate thirst center, it will stimulate conditional reabsorption of water, so loss of water is not there. Okay, loss of water is not there. Don't duplicate. Okay. Students, last mechanism is ANF. It is very small, okay. Hardly given in your textbook actually, only name is like much is not explained, but I will just give you a little more idea. ANF means your atrial natriuretic. Yeah, it will take DCT's and CT's help. Nandini, both. Okay. Thank you, Praveen. Atrial, okay, atrial natriuretic factor. Natriuretic factor. Okay, this is released by the heart. Okay, this is released by the heart. Clear? Okay. Now what is going to happen? Okay, here students, this ANF is actually opposite to the RAS mechanism. It is opposite to RAS and ADH mechanism. So I'm going to write here, it is opposite to RAS and ADH mechanism. That means it is not going to reabsorb water, it is not going to take in sodium, but it is going to give water and give sodium, lose it. Why? Because it could be because the person has drank a lot of water, it could be because the person has a lot of salt content, right? So ANF will work that way. Clear to all of you? It will work against diuresis and your natriuresis. So diuresis means it is going to lose water, right? Diuresis means it is going to make sure that water is lost. Okay. It is going to work against RAS and ADH only, will give you more idea. And natriuretic means it is removing your NaCl. NaCl removal. Clear? Clear? So, how does this happen? Like, why do they do this? What is the condition? When your BP is high, when your blood volume is high, when H2O is high in your body, water is high. In this kind of problems, when this kind of problems happen, they do this. They do this. Clear? Yes. Can I continue? Yeah, NaCl, a whole NaCl. Okay, I am only talking about Na. It's a whole NaCl. Okay. Is this clear to all of you? Yes. Now we'll do micturition. Okay, micturition means voiding of urine. That means students, all of you wash your face and come quickly. Take off all your sleep. Take off all your sleep because I am feeling lack of energy with you people. Kya ho gaya? Did you all like the session? NEET, did you like? Friends, did you like? Your names are like some cartoon. Okay. So, students, voiding of urine. Okay. So, micturition means voiding of urine or removal, that khali we tell, no, to remove the urine out. Okay, to remove the urine out. Now, how is this going to happen? We are going to study the pathway. So, I told you I can only call it urine when it reaches the collecting duct, correct? I told you that I can call it urine when it reaches the collecting duct. So, from collecting duct, how is it passed on? From collecting duct, it is going to go to duct of Bellini. From duct of Bellini, it is going to your minor calyx. Students, collecting duct, all of you remember, okay, how it is, right? How it is. Duct of Bellini. From there, it will go to the minor calyx. Okay. It is Bertini, no? Duct. Okay. Minor calyx, major calyx, renal pelvis. I want all of you to go see your diagram, then only you will understand this pathway. From renal pelvis, where it will go? Ureter. Okay, ureter. Students, no more break, I am sorry. No more breaks. Okay. Urinary bladder. From urinary bladder, it will go to the urethra and it will go out. Clear? Okay. Okay. Is this clear? Collecting duct. All of you know that I can call it urine only when it is a collecting duct. Collecting duct, okay. From collecting duct to duct of Bertini, from there to minor calyx, major calyx, renal pelvis, ureter, urinary bladder, urethra. From urethra, the urine is going out. Going out. Got it? Simple? Simple, no? This is how the pathway is. I taught you this pathway just before some time when I told you the flow. I told you I will teach you this. Okay? Yeah, duct of Bellini, no? I think I got confused. Duct of Bellini. I think I wrote. Yeah, this one. Don't think it's there in your duct of Bellini. I got confused with Bertini and. Okay. Students, now what is going to happen? Like I informed you when I was teaching you urinary bladder, stretch receptor is going to, I'll write SR, okay, stretch receptor is going to send message to the CNS that the urinary bladder is full, correct, the urinary bladder is full. Now what will the CNS do? It will initiate contraction of the urinary bladder, it will initiate contraction of urinary bladder. Now tell me which is the muscle that is going to help in contraction? Detrusor muscle. Okay, detrusor muscle. Okay, thank you Ashwati. Detrusor muscle. That detrusor muscle will contract and sphincter muscle will relax. Sphincter muscle will relax, causing urine to come out. This I told you sometime back also when I taught you urinary bladder, I told you this, correct? I told you this, remember? Yes. So, SR is stretch receptor, stretch receptor will send message to CNS, CNS will initiate contraction of the urinary bladder, that is your detrusor muscle, that by that way the sphincter muscles will relax and urine will come out. Clear? Easy? Easy? I explained this before, that is why I am going a little fast. I hope it is clear. Okay. Now, okay. Now, accessory excretory structures. Okay, students? Micturition clear? Any doubt? Any doubt? The yellow color in the urine, it is because of urochrome. That also I will write it here in case MCQ comes. Yellow color of the urine, okay, it is because of urochrome. The color is given because of urochrome. Clear? How much liters of urine is going out? Urine around 1 to 1.5 liters. Okay, 1 to 1.5 liters. Okay, and how much urea will be there in that urine? Okay, how much urea will be there in that urine? 20 to 30. 20 to 30 grams. Okay, 20 to 30 grams. Clear? Yes. Okay. This general information I have given you. Now, accessory excretory structures. Students, accessory excretory structures means there are structures other than the kidney who is helping to emit at least little of the nitrogenous or some waste outside. Correct? Some waste outside. Okay. So, for that we need to write some things. Okay, I am just going to write because these things are not very difficult. Sweat. Oh, 1 second. Okay, sweat. Saliva. I am just writing whatever is there in your textbook. Sebum. Lungs. Okay. Sebum, sweat, saliva, sebum, lungs, liver also is mentioned, I guess. Students, check and let me know. I cannot remember so much. Okay. Yeah, that is what. Duct of Bellini is in the nephron, but it's not mentioned. We never learn about it. That is why even I thought I got confused. Okay, duct of Bellini is in nephron, duct of Bertini is in your whole medullary pyramid, the between gap. Okay, thank you Nandini. Okay, students listen, sweat, what and all we will remove. NaCl, okay, some amount of salt, okay, some amount of salt will be removed, urea and lactic acid, okay. Then saliva, what and all will be removed in saliva? Very less amount of nitrogenous waste, okay, actually very less amount of nitrogenous waste, okay, clear. Sebum, okay, sebum. Sebum will release some amount of hydrocarbons and waxes, okay. And waxes, okay. Then lungs, what will it remove? Lungs, what will it remove? CO2, okay, CO2 it will remove, right. It will also remove some amount of water, okay, some amount of water. I think in your textbook it is mentioned 200 ml, okay, 200 ml is mentioned, correct, per minute, per minute. Okay. Then liver, liver will secrete out cholesterol, steroids, hormones, okay, all this it will secrete out, clear. It will secrete out bilirubin, okay, bilirubin, cholesterol, steroid hormones. One second, students. Yeah. Yes, bilirubin, cholesterol, steroid hormones. Okay, and steroid hormones. Clear to all of you? This is just memorizing. You just have to memorize these things, okay. Finally, we are coming to an end of our story. That is last one, artificial kidney. So students, when we talk about artificial kidney, we have to know that what happens. Now I am going to answer the meme that some student was asking, okay. Right, some student was asking what will happen if I give both the kidneys. Students, what will happen? The urea content in the body will increase so much that you will get uremia. You will get uremia, which is maximum amount of urea in your blood. Now, the only way for you to get rid of that is by artificial kidneys, which are known as dialyzing, dialyzer. We use dialysis as a method to make sure that the urea is removed out, but it is not for a long run, okay. We cannot depend on dialysis because it is very expensive, and there are people who have to do it monthly, weekly, yearly, and it is definitely a big burden for the family. So what is the other solution? Transplantation. Okay, transplanting kidney is easy, and every person can live with one kidney if they have a very healthy diet, without alcohol, without unnecessary things. If they have a healthy diet, then one kidney is more than enough to live. Okay, got it? Cleared all of you? Now look here, to avoid uremia, we need to do dialyzing. Now I am not going to tell you everything, but just look at what I am telling you, only that much. This much is not there for you. Okay. So students, this tube that is going is called your cellophane. Cellophane. Can you see if I write like this? Okay. Cellophane tape. Okay, cellophane tape is used, and that cellophane tape is completely coiled, and I think I will only draw because this looks complicated. Okay, students, here only I will draw. So look here. Students, this is the dialyzing machine. Okay, imagine this is the dialyzing machine, and this is your cellophane tape. Okay, this is just an example. This is not the actual how it looks. Just now you saw how it looks. Now this is a cellophane tape completely coiled. Okay. Now in the cellophane tape, now imagine we are this surrounding, right? Surrounding, now this dots which I am drawing. Here there is no urea at all. This portion is called as your dialyzing fluid. Dialyzing fluid. Students, what is dialyzing fluid? This cellophane tape is put inside this dialyzing fluid is there. In the dialyzing fluid, there is no urea at all. Okay. Now, I am going to take blood from a person. Okay. I am going to take blood from a person. This is the person. Okay. From this person, I have taken blood. This person's blood is full of urea. Now, you all know osmosis, you all know diffusion. Now, you tell me, will urea move inside or outside? Will urea move inside or outside? Pallavi, Ammu, Awkward Potato, all of you, did you like the session anyway? You're spamming, at least do that. Okay. Since you're spamming. Okay. So, this dialyzing fluid will not at all have any urea. Okay. So, this person's blood will enter. As this person's blood is full of urea, urea will start coming out. Urea will start coming out to the fluid. Clear all of you? Okay. Now, when I am adding this blood, I am going to add heparin. I am going to add heparin, which will make sure that there is no clotting of blood that is happening. And when I am taking this blood out after purification, now what has happened? All urea has gone out, right? So, now I will be easily able to take a purified blood. Now, I have taken it through the artery. I will take it back with the help of vein. Here, before taking this blood, I am going to add anti-heparin. Because body will already have heparin. So, I am going to put anti-heparin. Clear all of you? Yes or no? Yes or no? Yes, heparin is an anticoagulant. Okay, got it? So, there is a cellophane tape which is surrounded by your dialyzing liquid. Okay, it is there. Okay, dialyzing liquid. Clear? Now, this dialyzing liquid does not have any urea. Because the dialyzing liquid does not have any urea, the blood which is going from this person who does not have 2 kidneys will have completely urea. That urea will be removed out into this dialyzing liquid, and when I am taking in the blood with the help of artery, I am adding heparin, which is an anticoagulant, and when I am taking out the purified blood with the help of vein, I am adding anti-heparin. Okay. Clear to all of you? Is artificial kidney clear? Dialysing clear? See, this is the structure. From a person's artery, we have taken blood. Okay, blood is pumped continuously. Here, heparin will be added. That heparin will go through a dialyzing filter, that is your whole filter, which has no urea at all. After the purification has happened, it will be transferred again and taken back with the help of vein. MCQs will come from this portion. Okay, those who are ignoring this, I am sorry to say, you are losing your marks. Okay, and the process is called hemodialysis. Okay, hemodialysis. Clear to all of you? Anyone has any doubt? Was I too fast? Is the speed okay? Did you understand? I thought it was not that complicated, right? So, dialyzing or your using of hemodialysis is not a long term process. It is not going to help you for a long term. So, what is going to happen? Basically, we have to transplant kidney. So, when we are transplanting kidney, okay, we need to make sure that the kidney does not get rejected. Okay. So, that time we have to add your suppressant. Remember all of you, cyclosporin A. Okay, cyclosporin A, right. So, no, dialyzing fluid will never enter the blood. It will not enter the blood. Okay. Okay. Yes. Clear? Okay. So, students, look here, all of you, this, what do you tell? Your kidney transplantation is the only process. Kidney transplantation is the only process for a long term. So, what can we do? We can take kidney from our relatives, family members, anyone, okay, family members, anyone, we can take the kidney and we can be happy with that, even they can be happy with that. But we have to make sure that the kidney does not get rejected by our body, because we are taking kidney from another person, our body will think it is a foreign particle. So, we use an immunosuppressant. Like cyclosporin A. Okay. Then, students, renal calculi, all of you know this term, it is simply known as your kidney stone. All of you know kidney stone. If you don't drink water, haven't you heard parents telling that you will get kidney stone? You would have thought it is some stone that I swallowed and it would have gone all the way till my kidney. So, it is the stone which is a mass of crystallized salts which are formed of oxalates, phosphates, etc. Okay, that will get clustered in the kidney, and whenever a person moves and all, the stone also will move, causing them very, very bad pain. Okay, so if you don't drink ample amount of water, so I'll drink. If you don't drink ample amount of water, you will get kidney stone. Okay, I just protected myself from kidney stone. That's all. Renal calculi, other word is kidney stone. That means stone which is accumulated by crystallized salts like oxalates, phosphates, etc. etc. Okay. Now, glomerulonephritis, basically inflammation of your glomeruli of the kidney. Student, this is a very rare condition. Not everyone will have this condition. But in case your glomerulus get, swollen or anything, that can lead to glomerular nephritis. That means the filtration, glomerular filtration will not take place properly. Yes, we have reached the climax. Can we have a dab? Come on. Can we have a dab? All of you have the dab emoji, no? Not bad. So many students stayed back. I'm so happy. Students, quickly dab in the chat box. We have completed one big chapter. Yes, Ajmi, very good. Ajmi, very good, very good. Yes, wow, that's a dab. Everyone will think, why are they dabbing, no? It's because we completed. We completed with lot of hard work. Yes, how did you all feel? Those who were new to the session, do not forget to comment. Write down your scores. You would have got 6 marks, right? 6 marks a guarantee. So, write down your scores without fail, and also tell how did you feel the session. If you are new to the session, I will read all your comments. I will reply to all your comments. Okay. And do not forget to join my Telegram channel, which is TME/Gopika_NEET. Or you can just search Biology by Gopika, you will get it. Okay, those who are new to the session, do not forget to comment. Okay, and let us know if day 5 is completed. And if you want to join Avengers batch, I am in the batch. I am the wildcard entry. Okay, Big Boss, like that wildcard entry happened. So, you have to put my code Gopika to join the batch. Okay. So, if you want more wildcard entry, you have to keep using my name, so that I will get wildcard entry into batches. Clear to all of you? Yes? So, do not forget they are coming to Chennai. So, if you want to see them in Chennai, quickly fill the Google form and have a peaceful good night, students. Good night, students. Love you all. The session was amazing. The amount of support that you people have given is immense. That is the only reason I am able to stay without lunch, without eating anything, without drinking any coffee. I am able to stand in front of you is only because of the energy that I saw in the class. Okay, I hope the session was amazing. Those who joined newly to my session, do not forget to join me on my Telegram, because I would love to see you there. Okay. So, good night, students. Tomorrow at 11 o'clock, my special class link is pinned in the comments as well as on the description. Join me. We will do an amazing session of MCQs. Okay. So, see you all. See you all. Good night. Good night. Lots of love to my beautiful students. Take care. Now, I will run back to my home and have dinner. Okay. So, do not forget to comment how you felt. Do not forget to comment how you felt, how the session was. Okay. I know little here and there I rushed, but I hope I did not make any loss. Okay. And all your notes I will be posting. Okay. All your notes I will be posting. Clear? Thank you so much, students. Take care. Love you. Good night and sweet dreams. Thank you so much for your lovely messages. All of you who completed day 5, proudly put in the chat box that you completed day 5 on the comments once the session ends.